Hi, welcome to day 2 of our Neurotechnologies event, current developments, applications and ethical issues. We've got a great lineup of speakers today, and my colleague Marija Petrinovic will introduce them shortly. But for those of you who were unable to attend day 1, boy, you missed a lot. We had a fantastic overview of the neurotechnology landscape from Charlie Winkworth-Smith, who is a knowledge transfer manager, and the technologies emerging out of the UK just look amazing, but also where we sit compared to the rest of the world was a fascinating insight of how much headroom we have here in the UK to grow in this area. Michael Nietzsche really gave us a tour de force lecture on what transcranial electrical stimulation may be doing in the brain. And then we had a series of more clinical talks by Paul Shotbolt on functional neurological disorders, Lucia Valmagia in using virtual reality in mental health, and Ashkan Kamars on deep brain stimulation. We then moved more cellularly in the second half, focusing on stem cell based models for treatment and also nanoparticle delivery of treatments as well, ending really in a fantastic keynote from Viviane Tabar, covering the whole approach from the discovery to the application in surgery. All the talks that don't contain confidential patient information or risk of confidential patient information will be uploaded onto the King's YouTube channel after the meeting is finished, and all those who are registered will receive an email with the link in. But do look out for it on the King's webpage. So we're up for a second day with fantastic talks as well. And now I'm going to hand over to Marija, who has really led the development of this programme for the Research Innovation Committee at the IoPPN. Marija, over to you.
Thank you, Mitul. Thank you all of you in our audience for joining us today again or for the first time. If you, as Mitul said, if you have missed our first day, so yesterday's sessions, please do watch them on YouTube because they were really, really interesting. So we are continuing today with these super interesting talks and discoveries in the fields of neurotechnology, and we will start today with our keynote speaker at Boyden. We will continue with a couple of short talks, and we will end conference with keynote being delivered by Dustin Tyler. After that, and after the break, we will actually finish the conference with a panel discussion on the use and mostly actually ethics of the use of neurotechnologies in mental health and neurosciences and in society in general. But without any further ado, I would actually like to now announce our first speaker today, which I'm sure doesn't need any special announcements. His last name, I think everybody is well known and household names for all the neuroscientists. So at Boyden, Eva Tan Professor in Neurotechnology at the McGovern Institute and the HHMI in the USA. He's also a professor at the Departments of Brain and Cognitive Sciences, Media Arts and Sciences and Biological Engineering at the MIT. And he will tell us something about tools for analyzing and repairing the brain. Ed, thank you for joining us today. Much appreciated. We are looking forward to hear your talk.
My name is Ed Boyden, and I direct a group at MIT that works on tools for analyzing and repairing the brain. And we try to really confront the difficulties of the brain head on. The brain is so complicated, both in terms of spatial and temporal complexity. Brain cells are enormous objects, centimeters in spatial extent, but the wiring of the brain and the molecules along those wires are nanoscale and organized with nanoscale precision. Brain processes can take place over months or even years, whether it's the erasure of a memory or the progression of Alzheimer's disease. But the high-speed dynamics of the brain happen in seconds or even milliseconds. So I sometimes say that our group's job is to build tools to help brain scientists cross space and time. Today I'll tell you 3 short stories about technologies that we've developed which are in widespread use in neuroscience. Some of the newer ones are just starting to get out into the field. Some of the older ones are now in use by thousands of researchers. And all of these technologies really try to confront head-on the spatial and temporal complexity of the brain. Let's start with mapping the brain. Again, brain cells are large, but their connections and the wiring is nanoscale. Many pioneers have developed methods of nanoscale imaging, such as super-resolution microscopes or electron microscopes. But these are all difficult to apply to large 3D objects and can require expensive equipment. So in our group, we often think, what if we do the opposite of what everybody's doing? Rather than zoom in with a lens, which is how people have imaged biological specimens for literally 300 years, what if we physically make a biological specimen bigger? So the idea we came up with is to take a preserved biological specimen, like a piece of brain tissue, chemically synthesize a dense spiderweb-like mesh of swallowable polymer, basically the stuff in baby diapers, anchor key biomolecules or labels to the polymer, soften the specimen, and add water. The baby diaper polymer will absorb the water and then pull all the biomolecules or labels apart from each other. So in 2015, we announced that this is possible. This is work done by then grad students Fei Chen and Paul Tilburg in my group. Panel B is a small piece of the mouse brain before we expanded it. Panel C is the same piece of mouse brain tissue, about a day and a half later, and we expanded it by 100 times in volume, about 4.5 times in each direction. The polymer, shown in the upper left in cartoon form, is very dense. The spacing between polymer threads is just 1 or 2 nanometers, smaller than many biomolecules. And when stretched out, it attains the extended configuration shown in the lower left. We showed that we could use this strategy, which by design and by validation is even and precise down to the nanoscale. We could use this strategy to look at brain circuits. So here on the left, we're zooming in on the brain. This is a mouse with YFP in certain neurons. You can see the cortex and the hippocampus where my mouse is pointing. Each white square is zoomed in to the picture below. The color code is shown on the left. These are proteins being decorated with fluorescent antibodies. The images on the left were taken with a regular old confocal microscope. So very quickly we hit the resolution limit because light has a finite size or wavelength. The right-hand side is the same specimen, same fields of view, same microscope, but after we expand it. And I hope you can now see that we can very precisely detect things like synapses. The vague purplish blobs on the left are now clean blue and magenta domains on the right. And if you look at the distance between these presynaptic proteins shown in blue and the postsynaptic proteins shown in magenta, the distance is exactly what you would see with storm microscopy, as demonstrated by Xiaowei Zhuang and Catherine Dulac many years ago. But now you can do the same measurement on a regular old conventional microscope. That was visualizing synaptic proteins. You can also visualize fluorescent proteins. Here we are using viruses to express different fluorescent proteins in different cells through stochastic labeling. So some cells are blue, some are green. They have different fluorescent proteins expressed in them in combinations. This is the mouse hippocampus. If we zoom in with a regular microscope, we quickly hit the resolution limit of that microscope, as shown in the top middle. But if we expand, now we can resolve the fine wiring of the brain. And so many people are now using some kind of color code plus expansion to look at neural circuits and to understand how they change during natural or disease processes. So far, everything I showed you is looking at proteins, but you can also look at other biomolecules. We can anchor nucleic acids to the polymer and expand them away from each other as well. Oswasi joined the team, and we here showed that we could expand proteins and RNAs away from each other. You can see yellow fluorescent protein in green, and the RNAs are post-expansion labeled with fluorescent probes and are shown as purple dots. So we can map out different kinds of biomolecule, even in multiplex sets, in intact brain circuitry. So far, everything I've showed you is expanded by about 4-fold in linear dimension. But when J.B. Chang was in our group, we found that we could take a specimen, polymerize and expand it, form a second polymer in the space opened up by the first expansion, and expand it again. So you can expand over and over and over. Deblina Sarkar, Jinyun Kang, and Oswasi, with great help from Margaret Schroeder and Amuchi Eminari and Christina Kitko, developed important versions of this as well. we showed that we could anchor and expand proteins away from each other with all off-the-shelf chemicals, which had an additional cool effect. By separating proteins from each other, we could get better labeling. The top part of this cartoon shows the green and red antibodies trying to bind to densely packed proteins, and they can reach the outside of a protein complex. The lower half of the cartoon shows that after you expand proteins away from each other, now suddenly the yellow and blue antibodies can make their way in. So suddenly invisible proteins can turn into visible ones. We wondered, could we use this to visualize synaptic proteins, which are jam-packed together? Well, in panels B, C, and D, you can see examples where synaptic proteins like calcium channels, shown in B, and pre- and postsynaptic proteins, shown in C and D, are almost invisible when stained pre-expansion, shown in yellow. Yes, there's not much yellow to be seen. The purple, though, is the same specimens, same antibodies, but just restained after we decrowd the proteins away from each other. And now you can see much more detail. Not all proteins are crowded. Panels F, G, and H show some proteins where the yellow and the purple look more similar. But we were very excited now that we can start to really map out the fundamental building blocks of brain cells down to almost single molecule precision. Working with Li Wei Sai's group, we looked at Alzheimer's disease. This is a 5x FAD Alzheimer's model mouse. We wondered, amyloid plaques are a hallmark of Alzheimer's disease. They're very densely packed protein aggregates. If we expand them, can we see more detail? Here again, yellow is pre-expansion staining with an antibody against A beta 42, and purple is the same specimens, same field of view, same antibody, but just stained after expansion. I hope you can see there's a lot more detail in the purple than in the yellow. In fact, we even saw these mysterious, almost periodic structures where amyloid form these little dots along axons. We saw this with multiple different antibodies, and we never saw these amyloid dots in wild-type animals. Of course, you can look at other kinds of tissues from other species. When Yongxin Zhao and Octavia Bricura worked with our group and the groups of Astrid Wines, Andy Beck, and others, we looked at human tissues. On the left are normal, and on the right are cancer-containing tissues from prostate, lung, breast, pancreas, and a whole variety of other organs and tissue types. The color code is shown in the upper right. So to summarize the first half of this talk, we've discovered that we can physically magnify objects and preserve nanoscale information. It's easy to do with all off-the-shelf chemicals. And recently, the 300th experimental paper or preprint, not like a review piece, an actual experimental paper or preprint, came out doing an expansion process. It's being applied to the microbiome, to human kidney biopsies, to the fruit fly brain, to motor proteins, to stress granules, and the list goes on and on and on. All of our protocols are on our website, expansionmicroscopy.org. And we used to host lots of people at workshops, but with COVID, of course, we can't do that. And so we recently posted a step-by-step photographic tutorial on the website so anybody can learn how to do expansion. After all, biomolecules are nanoscale and often organized with nanoscale precision. Mapping out the 3D architecture of cells and tissues with nanoscale precision will open up many frontiers into how biological mechanisms are implemented and how they go wrong in disease states. So that's the first half of the talk. And I think to transition to the second half, I should point out the major limitation of expansion microscopy, which is that it doesn't work on living things. It works great on preserved cells and tissues from a wide variety of species, including, as I mentioned, human tissue, but it's hard to see how the expansion process would be compatible with a fully living state. And so in the last half of the talk, I would like to mention, with a focus on more recent areas of our work, what we're doing to control and look at high-speed dynamics of living systems in the brain. Let's start with optogenetics. And I won't talk too much about this because it's already pretty well known and widely used, and much of the actual molecules that are in use in neuroscience were published in 2010 or earlier. Optogenetics is how we control brain cell electrical activity using light. This started when Carl Dijsselroth and I were both students at Stanford brainstorming, how can we just control brain activity very precisely? We just started brainstorming through all the laws of physics, magnetic fields, mechanical force, and we decided light could be the best because light can be aimed at cells. You could bring light into the brain using probes like optical fibers. And the next question then became, Well, how do you make brain cells sense light? I became really fascinated by a class of molecule known as microbial opsins. These are membrane proteins, like those shown in the lower left, that are found in the membranes of single-celled microbes that live in water, like the salty water shown in the upper left. The first to be found was a light-driven proton pump, published in 1971, which converts sunlight into chemical ion gradients in single-celled microbes. A decade later, several groups found light-driven chloride pumps, which also convert sunlight into chemical stored energy. And then around the turn of the century, several groups working together found light-driven ion channels. Being channels, of course, they don't store energy, they can dissipate energy, but they're used by single-celled algae and other critters to sense light and use it to control different processes. So for example, single-celled algae use it to swim around and navigate in bodies of water. Flash forward to the current day, what we have found, along with many colleagues and collaborators, is that members of all three of these classes of molecule can be identified that are safe enough, fast enough, and powerful enough to be expressed and utilized in neurons. So when Brian Chow and Xue Han worked with me, we showed that certain light-driven proton pumps could be genetically expressed using viruses or other transgenic means to enable neurons to be controlled by pulses of green or yellow light. By pumping protons out of a cell, you could shut it down and figure out what it is needed for. In the middle column, Xue and later Amy Chuang found that certain light-driven chloride pumps could be safely, powerfully, and usefully expressed in neurons. And then we can use orange light, or as Amy found, even red light, which goes deeper in the body than other colors of light, to shut down neurons. So these are ways of silencing neural activity. On the right-hand side, light-driven ion channels come in several flavors, but the first that we expressed in neurons was a light-driven cation channel. Carl Dijsselroth and I collaborated together to put this into neurons and found that blue light could activate neural activity. And later, Nathan Klepecki in our group found a red light activated version. Again, useful because red light goes deeper than other colors of light. Now, literally thousands of researchers are using this strategy to activate or shut down neurons and figure out how they contribute to behaviors or pathologies. People are discovering patterns of brain activity, for example, that might help clean up different kinds of pathology. My collaborator Li Wei Sai, for example, has found that certain brain rhythms discovered with optogenetics in her lab might help clean up pathology of Alzheimer's disease. And that inspired some non-invasive strategies that might help deliver to the brain with movies, for example, to drive the same brain rhythms to clean up Alzheimer's pathology. Recently, a European team found that the molecule crimson, which I mentioned earlier, this is Nathan's molecule that is a red light drivable activator, can be expressed in the human retina of a blind patient and help restore partial vision function. So optogenetics is both widely used to make discoveries about how we could heal the brain, and even recently is starting to be used in people directly. That's all I want to say about optogenetics, because again, it's a fairly mature field at this point, but I do want to note how lucky we were. These molecules out of the box were safe enough, powerful enough, and fast enough to enable the control of neural activity with light. And that's a great segue into the final part of the talk. Because the flip side, watching the brain in action, we have not been so lucky. You can't find molecules that just out of the box from nature automatically allow you to see a signal in the brain. And so we thought, if the natural world won't provide these, let's evolve them in the lab. When Erica Jung and Kirill Pyakovich were in my group, we built a robot to do directed evolution. Take a gene and make many mutants. Some are better and some are worse for any goal. You can express them in cells and then use a robotic arm to pull out the cells that have the mutants that are better for your application. We wondered, could we make a fluorescent voltage indicator? Basically the opposite of optogenetics. Starting with Quasar 2, a molecule from Adam Cohen's group at Harvard, we did 2 rounds of directed evolution and made a molecule that we called Archon. This is some work that we collaborated with Xue Han's group at BU to express Archon in the live mouse brain. And then by shining red light on the mouse brain using the microscope in the upper left and collecting the red and infrared light onto a camera through the microscope, we can image the voltage of neural activity. So these traces on this slide look like electrode traces, but they're not. They're being picked up by a microscope. So we were able to make a fluorescent voltage indicator that was useful for picking up neural activity in a wide variety of brain regions, even from many neurons at once, in awake, behaving mice. We're very excited that our robot might help optimize lots of different fluorescent indicators. Of course, we're not the first to make fluorescent indicators. There are hundreds of them out there. This is a screenshot from Jin Zhang's database at UCSD. People have taken fluorescent proteins from coral and jellyfish and connected them to all sorts of sensor domains to pick up calcium, cyclic AMP, protein kinase A, and the list goes on and on and on. We wondered, wouldn't it be great to image many signals at once? Because then you could see how they relate to each other. That's a difficult thing to do, though. Shannon Johnson and Cheng-Yang Lingu and my group, we worked together to come up with a strategy so that anybody could image many signals at once in a living cell to see how those signals relate to each other, how cells compute, so to speak. The idea we took forth was, what if we could put indicators of one signal at one random set of points, here labeled 1, and indicators of a second signal at another set of random points, here labeled 2. While the cell's alive, each point will blink out a little movie of the signal that's being there reported. So maybe the label 1 points will tell you the calcium signals. The label 2 points will tell you the protein kinase A signals, just to pick 2 random signals. Later, you can figure out which signal is which because you can stain the cell after it's preserved with antibodies or other labels and figure out what the identity of the indicator is at each point. There are many ways to make this work, but our favorite way of doing it is to take fluorescent indicators and fuse them to self-assembling peptides. Then they randomly but stably cluster at different points in space. Each reporter also has an epitope so that we can stain it later with an antibody to figure out the identity of the reporters at that point. Here's an artist's conception of the idea. The green dots are when the cell's alive, and the false color image is what you would get after you stain the cell when it's preserved. In panel B is a neuron expressing GCAMP6, a well-loved calcium indicator. Panel C is the same calcium indicator, but now fused to self-assembling peptides, and you can see it forms discrete dots. We did tons of control experiments to show that the signals, the physiology, and the cell health are not compromised by expressing these clustered reporters. Amazingly, it's modular. We can take 3 different indicators, here for calcium, cyclic AMP, and protein kinase A, fuse them to different self-assembling peptides, and express them together in the same neuron. The green images are when the cell is alive, and the false color images are after the cell is preserved so you can stain and figure out which sensor is at each point. So while the cell is alive, you can record movies of all 3 of these signals. Here we look at calcium, cyclic AMP, and protein kinase A after we give a neuron a drug, forskolin, that drives cyclic AMP production. Now we can derive relationships between these signals. We noted in panel H that some neurons had slow calcium responses. In panel I, other neurons had fast calcium responses upon forskolin challenge. We wondered, protein kinase A is sort of the output of this network. Does it depend on the speed of the calcium? Amazingly, we could derive relationships between this. In panel L, you can see that the fast calcium neurons had strong protein kinase A output, and the slow calcium neurons had weak protein kinase A output. So we can relate the speed of one signal to the amplitude of another. We can add a protein kinase C indicator and look at 4 signals at once in a single living neuron. Here are some traces being recorded all at the same time from the same part of a cell. And we can add a 5th indicator. Here we add an ERK kinase indicator. And now we can look at 5 different signals in the same cell. So our hope is we can start to derive relationships between different signals. Cells, after all, have many inputs and many outputs. How do they compute inside the cell so that the outputs are appropriate given the inputs? So to summarize, we're very excited that we can build through evolution or through clever use of tools in new ways, new strategies for observing high-speed dynamics of neurons. We share all of our tools freely. Please visit our website, synthneuro.org, to learn how to access them or to get help on how to use them. And don't hesitate to email me, it's just my name, ed.boyden@mit.edu, if we can help in any way with your work. After all, we build these tools because we want to empower people to solve important biological problems. And with that, I'd be happy to take questions.
Thank you very much, Ed. And as I said yesterday, it is such a pity that we don't have any kind of a up here or something for the applause. So you just have to imagine it. But I'm sure that there will be a big, loud applause for this talk. Thank you very much. What we have noticed, before I take questions from the audience, what we have seen since we started this event yesterday is that new technologies, new tools for analyzing nervous system are now coming with such a speed that it is almost really, not almost, it's really difficult to even keep track of all of them. You know, it has never before, I think it was never before like that, like 10, 15 years ago, they were coming one after the other technologies, but now we have so many of them almost each month. We have few questions here from the audience, and I just have to remind audience to please put your questions in the forum. We will ask the questions after each talk, and you can even upvote them and so on. So first question that we got was about expansion microscopy. So one person asked, so how do you deal with the fact that maybe different organelles in the cell, they may have different expansion factor? You can expand them only as much, and also they have different refractory indices. So how do you deal with that when it comes to microscopy?
Yeah, great question. It's very important to realize that the polymer is extremely dense. The spacing between the polymer threads is just 1 to 2 nanometers, much smaller than many proteins and other biomolecules. So the expansion process is really about pulling molecules apart. And so we see expansion of the nucleus, we see expansion of mitochondria, endoplasmic reticulum, Golgi, and so forth, that is isotropic and even. In response to the second part of the question, can you repeat the second part?
So, different type of cells and different parts of each cell have different refractory indices when it comes to microscopy. So now when you expand everything, how do you deal with that problem?
Yeah, expansion solves that problem. When you expand an object 100 times in volume, it becomes 99% water. So it becomes completely transparent and, more importantly, even in refractive index. I'll give you an example. We published a paper with Eric Betzig that was on the cover of Science in January 2019. And he had built this lattice light sheet microscope that has adaptive optics that could compensate for refractive index and homogeneity. But after expansion, we found we did not need to actually use the adaptive optics. So we just were able to do regular light sheet imaging because of the extreme evenness of the refractive index. That paper also showed that we could look at mitochondria and lysosomes and other compartments of neurons. We also looked at myelination and also compared all sorts of different structures, both in the mouse brain and in the fruit fly brain.
And there was another question with respect to expansion microscopy and the technique. You probably answered it, but let us just repeat it maybe for the audience. How long, once when you expand the tissue, for how long can you store it?
Great question. So when you expand, you're using pure water or a low salt concentration, and we do not advise to store samples in pure water for very long. So in our group, most of the time, after we're done, we'll add salt back, and the buffers associated with the salt will then keep things more neutral in pH and prevent acidification, and that helps with long-term storage. There are also some protocols where we expand and then re-embed. So you can add water and expand to hydrogel, and then make a second charge neutral polymer that will then keep it expanded. And that can also stabilize imaging as well. Yeah, but in general, adding salt to shrink it back down will enable stable storage, at least as well as any fluorescent stain can be stored, right?
Yeah, exactly. Thank you. We got a question with respect to the Archon and the combination of that technology with optogenetics. How easy it is? Is it possible? Was it already done?
Yeah, many other groups are combining live imaging or perturbation with expansion. I'll just pick one example that comes to mind, but the Fitzpatrick lab had a paper in Neuron where they did optogenetics to look at synaptic connectivity, and then did expansion microscopy to pinpoint the dendritic spines where the synapses landed. And so that's been published in 2020, I think. Yeah, so it's a very feasible thing to do. In our group, we're doing a lot of work now where we are imaging neural activity and then seeking to apply expansion to the same sample so that we can connect cell types or cell anatomy to neuroactivity patterns. But others have already published such things.
Okay, we have quite a few questions basically asking the same thing. As you said, optogenetics is there already for some time, and it is quite well-established methodology preclinically. And there are some attempts to also bring it to the clinic, and you have mentioned one of the examples. But what is your view on actually using optogenetics for repairing the brain, or even before kind of damage happens, kind of to re-engineering the brain if necessary from the start? What are the challenges for bringing actually that methodology to the clinic?
Well, methodology, so maybe the analogy that one can make is, if you have a method of control, you still have to target it to a certain brain region or cell type. In other words, there needs to be a reason to control a specific cell type or brain region to help somebody. So I think the big challenge is, how do you determine what brain region to target or what cell type within a brain region to target? In practice, that's difficult because even in a mouse brain, the cell types might be different. In fact, we know that they're different from the cell types in the human brain. So I think there's two paths that seem to be emerging. One, which I mentioned, Li Wei-Sai's been doing, where she used optogenetics to discover that a certain brain wave in a mouse model of Alzheimer's could help clean up Alzheimer's pathology. And then the team under her leadership went on to show that you could cause the same brain wave by having mice basically watch a movie, see a flickering light, and hear a clicking sound. And now several teams are working on clinical trials of this kind of strategy to have flickering lights and clicking sounds to see if you can induce this brainwave non-invasively. The other is to have very clear science knowledge. So I don't think it's a coincidence that the first uses of optogenetics are in clinical trials for blindness. The inputs to the eye are fairly well understood, at least much better understood than much of the human brain. The flow of information, we know how it looks in the eye, and a lot is known about the cell types and their molecular recipes. So yeah, there needs to be a reason, right? We think we should put the optogenate molecule in this brain region and this cell type, and here's the evidence why. I think that's really the key. But I think that, of course, the other question is, these are genes from other species, right? From bacteria and algae and fungi and other critters. Will there be an immune response? Will there be, changes in cell physiology or health over time? And for that, the clinical data has to show it in clinical trials, which of course is what the European team that I mentioned is doing. So if the trials go well in blindness, then I think that reduces the risk a lot. But you still need to know something of the science, right? Where do you put the molecule in order to help somebody?
Exactly. Thank you. So this was actually, one question covering many from the audience. And there is another one coming in saying like this: how long does it take for the expansion to occur? Is it an immediate process, or does it take a few hours to occur?
The expansion itself can happen within a few tens of minutes. That's pretty fast. Typically, the polymerization will occur over, maybe a couple hours. Then you can soften it with a chemical treatment, and that can take, depending on how you do it, an hour or two if you use heat, or overnight if you use enzymes. And then usually if it's enzymatic, we'll come back the next morning, we add water, and then it'll expand within a few tens of minutes. But the whole process can be streamlined. Yongshin Zhao and he's now a professor at Carnegie Mellon. He did the human tissue work that I mentioned. His group and our group recently had a joint paper where we showed that for thin human tissue biopsies, the kind that are used in the pathology clinic, the whole process can be got down from start to finish to a couple hours. So there's nothing fundamental saying that it has to take a day. Although if you are having a process that takes several hours, it can be convenient to run it overnight just because you don't have to sit there. You can go home.
Okay, thank you. And one more question. We were asked, somebody asked in the audience, so what about combining, let me see what it is, tissue clearing, so kind of making the brain transparent with the expansion microscopy?
So the expansion, as a byproduct of the expansion process, automatically clears the tissue. In fact, I've been told by several people, I'm not an expert in this, but it's the most clear of any tissue, because you're not just evening out bumps in refractive index. By diluting everything 100-fold, you're basically setting the refractive index within 1% or so equal to that of water. So as I mentioned, Eric Betzig, who won the Nobel Prize for his work on microscopy, when they were doing lattice light sheet microscopy, which is sensitive to refractive index and homogeneities, found that they didn't even have to do adaptive optics. They could shine light from one side, take a picture from a 90-degree angle, and without adaptive optics compensation, could scan through the tissue. It's very, very transparent.
Excellent. Thank you so much, Ed. All the questions which are not answered right now can be published and maybe answered later, because we need to move in the with the conference with our next speaker. And Ed, we are looking forward to hear again your views on the use of neurotechnologies that you have just mentioned and their ethics in the panel. Thank you again for joining us today. And now we are moving to a talk from, this time we are moving across the ocean back to Europe, to Italy, where we have Alessandro Gozzi sitting in Rovereto, right? Right, Alessandro?
Yes,
so Alessandro
indeed.
is
yes.
At the Italian Institute of Technology in Rovereto. Actually, Alessandro, where exactly in Italy is that?
Rovereto is in the north of Italy. Is at the foot of the Dolomites somehow, or nestled in the Dolomites. So it's a very beautiful place, yes.
Very beautiful.
Very lucky, yes.
So very soon you will be able to go skiing, right?
Absolutely, hopefully, yes.
Pandemic permits.
Yeah.
Alessandro, thank you very much for joining us today. And Alessandro will tell us something about neuroimaging methodologies, but at a macro scale or mesoscale, how we like to call it. So he will be talking about MRI and how to use MRI to parse autism heterogeneity, because we all know that heterogeneity in autism is often referred to as the major roadblock for successful clinical trials. Alessandro, thank you. And this is now your time.
Thanks a lot. And thanks for having me here. So as you just said, in my talk, probably it will be a little less, or clearly a lot less technology oriented than what we did, the fantastic work we've just seen so far. Still, I mean, the objective here is to show how advancement in whole brain imaging across species, in my case, we'll be using fMRI that we applied also to preclinical models, can help really bridge clinical and preclinical neuroscience and somehow address questions that are possibly of clinical relevance. And so the research I'm going to present today has been strongly influenced by the result of a series of studies that appeared pretty much 1 decade ago or so, showing that in autism, very often there's a disruption in brain connectivity, as I said, as assessed with fMRI. So these studies, or there's an example here, but there were many, show basically that the brain in people with autism very often has got like a problem with synchronization, so areas are dysynchronous, okay? And so the result of this research has prompted a second wave of additional study employing neuroimaging methods in humans, aim to address perhaps a more important and more ambitious question, which is whether we can somehow see autism in the brain. In other words, here the idea is that perhaps if you just image a larger enough sample of patients, we can identify a single signature of network dysfunction that can be specific, that can define the condition, so autism, okay? And so these studies have occurred over the years. However, the results of this second wave of investigation have been pretty sobering, because as I show you here, there are of course many reports showing that functional communication and connectivity is disrupted in autism, but there are also studies showing exactly the opposite, that there could be like aberrant or over communication. And now there are also like this mega study we're trying to put everything together, perhaps indicating that we got somewhere in the middle, right? We got like areas that are hyperconnected, areas that are hypoconnected, and it is very difficult to make sense of these findings. And so, well, this result, of course, have prompted a discussion and controversy in the field. There are different ways of somehow interpreting these conflicting results. However, a prevalent way of interpreting or contextualizing these inconsistent findings is the one that is epitomized by this study here. So the idea that, there must be basically a signature of dysfunction that is specific to autism, and the reason why we not found it yet is because we not be using the right methods. For example, fMRI now is like under scrutiny because of this discussion about reproducibility of neuroscience and so on and so forth. And this is clearly a possibility, and as the discussion goes on as we speak, if you like. But however, the research I want to present briefly today was somehow designed to probe a possible, a second, equally plausible, in my opinion, explanation to this inconsistency. An explanation could be the one that I'm capturing in this slide here. The fact that perhaps if we've not found such a signature of dysfunction across patients is perhaps there are indeed patient populations that have like lower functional communication, as I said, the fMRI, patient population that have more, and perhaps a third category of people that have with autism that have like some sort of a mixed signature somehow. And this view is actually consistent with the etiology of autism, which we know is a spectrum, so it's very heterogeneous, and most importantly also with the result of genetics, right? We know that autism is a constellation of disorders that are strongly heritable, and the genes that have been associated with autism that confer risk to autism are now pretty much more than, well, 100, even more now, and none of this single gene risk or autism risk genes actually explains more than 1% of the cases. So there's a possibility that what we've seen is actually what we would expect to see, right? The question now was, when we started this study, is how can we rigorously, probe these hypotheses? How can be tested? Well, there are different ways. I would just illustrate briefly two. There's like a plan A, if you like, and a plan B. And both rely on the use of genetic as a key for an etiological deconstruction of autism. In other words, what we can do, we can, for example, identify, let's say our plan A will be identify people with autism that are etiologically homogeneous, to scan their brains with fMRI, EEG, or some neuroimaging methods, and see whether the resulting signatures of connectivity or network dysfunction are converging or diverging or different. And this is being done. However, it's going to take a long time because as I mentioned before, these mutations are very rare, and it is very difficult to find enough patients. And also, there's a big discussion about etiological homogeneity. And so, what we thought in the lab is, okay, how about we try to, think of a plan B? We try to switch this approach to a model organism, such as rodents, where we can somehow map simultaneously many mutations, but simultaneously as part of the same study, of course, and where there are, of course, also a number of additional advantages. For one, the fact that we have a clear control population, which is always a problem for autism. We can control very well also confounds that plague somehow human imaging or human research, such as environmental contributions or the problem of head motion and so on and so forth. And so what we did, we tried to somehow reverse engineer models, methods more than models that are used to map brain imaging in humans. So we implemented fMRI in rodents, and that's exactly what we undertook, actually, which we sought to do. And here, this is the only technical slide, but I won't spend much time here. But I want just to tell you that this is made possible by research that was pioneered in my lab, where we basically show that we can effectively implement resonance-aided fMRI in rodents, and to a certain extent, the functional organization of the rodent brain is comparable to humans. Of course, this extrapolation can be done with some caveats, but the general foundational principle of this activity is conserved. Okay, so let me get to the point of my study here. And what we basically did, we said, okay, let's try to recreate the genetic landscape or the connectional landscape, because here with these methods we wanted to map functional connectivity. And so we gathered together a large enough number of models. This was done in collaboration with our friends in Zurich. And then we, for each of these models, we mapped functional connectivity. And what I'm showing you here is a low-dimensional representation of the results we got. across mutations and across mice. So each dot here is a single scan, okay? And here there are two important take-home messages or points I would like to deliver. The first one is that if you look at the individual dots that have the same colors, they're pretty much clustered, which is nice because this is something you would expect, right? So we can somehow, you know, reverse engineer the mutations here if we wanted. That wasn't the goal of the study. But what is important, however, is that by showing you this, the point that is really emerging is that this method is, if done after or under control conditions, is not as noisy as it is depicted. So fMRI can be actually pretty precise and accurate. Okay. And the second important point, perhaps the most relevant for the biology here, is that if you look at the distribution here, we have a bit of a landscape. We don't have like an homogeneously distributed cloud of points. This is really arguing against a single signature of dysfunction. And the same results are perhaps more apparent, more evident here when I'm showing you for each mutation the corresponding connectivity difference. So blue means reduced connectivity. Each brain here, this is like a mouse brain, and it is reporting what we found in a single mouse line and or mutation. And as you can see here, there are indeed convergences. For example, these 4 mutations all produce hypoconnectivity in these midline areas, which are like associative areas, the mouse brain default mode network, that's the name of it. However, as we zoom out and we add more mice, we have, for example, here a shift towards posterior regions. We have mouse models and mutations or etiologies where there isn't much happening in the cortex, there's more subcortical. And what's more interesting and telling is here when we switch from blue to red, red meaning overconnectivity. And so what is very apparent here is that first, etiological variability is a key determinant to the connectivity alterations we have in humans. That's really our hypothesis, and here it's very visible, it's very apparent. The second important point then is that it is unlikely that, at least using fMRI, we'll ever see a single signature of dysfunction associated with autism. But also there's a third important point that some of these mutations, however, converge to disrupt certain circuits. And that's really what I would like to prove here and further probe in the last few slides that I have in my presentation. I want to show you how we can perhaps make use of these convergences we found with a single and simple example that we recently published. The idea here, okay, can we make use of these clusters and try to relate to specific traits that happen, for example, or that are pathologically relevant for autism. And the example I'm going to show you here is a simple study when we try to understand what could be the connectivity profile associated with a trait, a pathological trait that has been often found post-mortem in tissue, brain tissues of people with autism, which is represented in this beautiful picture here, which is an excess of dendritic spines, an excess of synapses, okay? And the questions were two. So what happens when we have this excess of synapses in terms of functional connectivity? And second, can we decode the resulting connectivity signature in clinical population? Given that we're using fMRI, right, we can somehow translate across species. So in order to make this study possible, we were able to leverage important work that was done by the Salzer lab at Columbia. Basically, what the lab showed is that in this tissue, post-mortem tissue, very often is increase of synapses associated with overactivity of a molecular pathway. This molecular pathway is the mTOR pathway. This is probably a little too technical for this talk. What's really important, however, is that this molecular pathway is a point of convergence of many mutations of autism, and also it can be very well modeled in the mouse because it's a pathway we know quite well. And so that's what we did. We used like a mouse line that recapitulates this hyperactivity in this pathway. We check that there are more dendritic spines with respect to control conditions, which is like a bit of a sanity check. And we measure the corresponding connectivity signature, which is interesting because we find basically that where there is more connectivity, well, when there is this trait, there's more connectivity, and this is occurring in frontostriatal insular region. So it's a very nice loop that we know very well, and also to be important, we know to be important for autism or, for example, stereotypies. What's nice about this approach, this bottom-up approach, is also that we can probe the mechanistic specificity. So we said, okay, we want to link this to this increased number of spines. We can somehow use a drug, for example, your rapamycin during development, to normalize dendritic spine numbers and see what happens to connectivity. And as you can see here, this is connectivity. Red is like hyperconnectivity. Blue is the opposite. It is the rescue. And as you can see, really, we have like a total rescue, which here on the right can be related also to spine density. That is, when we normalize spine density, so that we reduce this excess, we remove this excess of spines, connectivity normalizes, which is nice because we can link these two phenomena occurring at two different scales. Okay, so I have like a couple of slides to conclude. And the point here, which is very important, is how can we make use of this when translating findings across species, right? Because that was really the goal of this, so try to inform clinical research. Well, we know that this trait, this excess of synapses, produces overconnectivity in this specific circuit, which is, as I said, cortical frontal striatal. What we did by using machine learning, now we'll spare you the details, we kind of decode the same signature in a large database of scans in humans. This can be done because we know where to look for, right? And as you can see, the circuit looks the same. However, what we went further and we tried to establish a molecular link, and the way that can be done, this was the input from my colleague Mike, was basically by comparing this circuit, this aberrant circuit involvement with patterns of gene expression. For example, in the case here, we found like pattern of post-mortem gene expression of genes that are interacting with TSC2 and mTOR, which are these two genes that are important for this excess of dendritic spines. And that's nice because this establishes some sort of a molecular signature to this pattern. But the last thing I wanted to explain is this, and it is very important because it really closes the loop. So far, I told you that our mice studies suggested there could be different signatures in humans, and that's exactly what we found. When we just cluster, right, this circuit, these connectivity circuits in this heterogeneous population, we found that this can be clustered into 4 different subtypes. Only one of which is the one labeled subtype 2 here, is exhibiting this enrichment in this gene expression. All the rest are different, and if you look at them, some of them are really divergent, like this over-connected, blue is like hypo-connected, and something in between. This really closes the loop because it shows that we can, identify with this bottom-up approach signatures in model organisms, and we can use this information to decode clinical heterogeneity. So I think my time is up. These are like the 3 take-home messages. I think this method can be used to unravel autism connectopathy, and this also can be extended to other disorders. And what's perhaps important, we provide evidence that this heterogeneity can explain heterogeneous connectopathy in clinical populations, but this can be used to our advantage, why we can perhaps use these to identify neurosubtypes, okay? So etiological homogeneous subtypes. Thank you for your attention. These are like all the funding agencies and the people in the lab that help make this possible.
Thank you, Alessandro. Alessandro, we have lost your camera feed. We cannot see you.
There I am.
I hope everything is okay with your camera.
Yeah, I think I just have deactivated this somehow. Can you see me now?
Let's see. Yes, I can see you now. Alessandro, we have a few questions, but we're slightly over time, so I will probably need to cut a few. So first question is actually coming from Mitul. So is the temporal architecture of the RS signal in mouse similar to human? And how does cardiac and respiratory noise affect the analysis?
Yes, the temporal structure is the same because these are still infra-slow signals. So they are pretty much exactly the same. There is no scaling across species for this signal. So it's got like 1/f distribution. And the peak is around 0.03 hertz in terms of power. So we find that is something that is totally conserved across species, which is pretty interesting. Regarding cardiac and respiratory, this is something that we can control very well. And under these conditions, the variance explains is really marginal. So that's not a major confounder, I would say here.
Thanks, Alessandro. I just turned my microphone on to say hello.
Hi, Mitul. Yeah, good to see you. Well, virtually at least, yes.
Excellent talk. If Marija will allow me, I'll ask another question, but I'll let other questions. Is that all right, Marija?
Yeah, sure. Go, Mitul. So
I really like the Pagani paper where you looked at the subtypes and the gene expression. Now you use the Allen Brain Atlas, is that correct, for the gene expression? I'm just wondering what you think about those methods, because it's quite a low resolution atlas, really, because obviously there aren't that many participants in it yet, and it's been incredibly useful. But do you really have the resolution to separate the subtypes really reliably?
Yes, because in such case, I fully agree that this is like as data set is still. It's only have 6 participants and so on, but that's something they were addressed, I think, very rigorously in the paper. But regarding your question, resolution, this is not an issue for functional connectivity, because the functional connectivity network we are interested in, and these microcircuits are very much extended, and they largely exceed the resolution that is required. So these are really like a large-scale phenomena. This might, however, be a problem for other kind of approaches. I agree. So, but I have strong confidence in the fact that this was not that our research, or our research question was not affected by or limited by resolution issues.
Great, thank you.
Alessandro, one last question, and probably a question that I really don't like to get, but I need to ask you because it is in the feed. Basically what you're doing now, it's kind of reverse imaging genetics. Years ago when everything started, we first have genetics, then we image the mice, but now we can go, as you said very beautifully, in the clinics, you can use imaging to maybe find out what the possible genetics is. But in preclinical research, and this is the question, we use anesthetics or sedatives to keep the mice still. So how do you deal with that, and how does that compare with with clinical studies?
Yes, well, that's an interesting question. A lot of this work, well, actually, pretty much all this work was done under light sedation. However, we are releasing, and there's already a bioarchive I can point you to out a paper where we show the sedation and anesthesia. Well, light sedation we use, and awake condition, we have now a protocol for awake had fixed mice, only affect the dynamics a bit in this. So basically the sequence of states had happened, but the overall network architecture, as long as the anesthesia is light, is pretty much preserved. That's something we addressed rigorously, so I can point perhaps reader to this paper that hopefully should appear soon also in publication. And that's something, by the way, that is very much in line with research across species. In monkeys is the same, in humans is the same. So the overall network structure is the same. Well, it's pretty much preserved, I would say. And what changes is the dynamics. And the dynamics, of course, it's another dimension we can use in the future. So we have to be careful there. But what we did here was like time averaging. And so we are confident that whatever impact there was there was, let's say, marginal and not very relevant to our findings. In fact, we're able to translate across species.
Thank you very much, Alessandro. And we need to move on. We are slightly behind the time. Thank you very much for joining us. And I'm sure that this was a very, very interesting talk for our audience. Thanks again. And now we are moving forward to a very interesting and exciting talk, especially for me, probably because I started as a PhD student working on spinal cord injury. So our next speakers, or a speaker, because one is not present today, unfortunately, is so Abigail Connor. Abigail, are you with us?
Oops. Yeah, hi, sorry. I'm just
okay, excellent. You're here. So
yeah.
Okay, sorry, I didn't see you. That's why I was just checking. So Abigail Connor and Stephanie Abramides, who unfortunately cannot join us today, as undergrad students at King's, they actually won a gold medal, and I really have to say this. They won a gold medal for the best therapeutics project in a competition run by the International Genetically Engineered Machine Foundation And their project was a new potential treatment for people with spinal cord injury. So we will hear more about that project in the next few minutes. So Abigail, thank you very much for joining us today and for sharing this really exciting news and your ideas about novel treatments for spinal cord injury patients.
Thank you. Thank you so much for the invitation. I'm really excited to be here. I am having some trouble sharing my slides. Can everyone? No, that's not right.
Abigail, if you share the screen you was just sharing and then go back to your presentation, it will come up. You had it full screen for a second.
Is that work? Okay. Is it possible to share it in presenter view at all? I'm trying to. Could I get permission to do that? I'm not sure how to.
You've only got one screen. Would you like me to share so you can see your notes?
If you don't mind, yes. I have rearranged the order a little bit.
Oh.
But that's okay. Okay. Right then, let me just present from here. Okay, so I'll just tell you when to, I guess, click to the next slide. Then I'm sorry, I thought I could like present it in presenter view, but that's quite okay, I guess. So yeah, good morning or good afternoon, wherever you may be in the world right now. I would like to say, first of all, thank you so much for the invitation. I'm super glad to be here, and I'm super grateful for the chance to speak about some of KCL's iGEM team's achievements over the past 2 years. So as Maria mentioned, my name is Abigail or Abby, and I'd like to welcome you to my presentation on Renovate Therapeutics, an organization founded and run by students dedicated to advancing treatments for spinal cord injury through the convergence of bioinspired materials and synthetic biology. So I have been, I've mentioned this term iGEM a little bit, and I think it's rather pertinent that I define exactly what this means. So kind of before I get too much into everything, I think it's important that we acknowledge that all of this work and all of the research that I'm going to be discussing in these next few minutes was all carried out by undergraduate students as a part of oh, this is really annoying, as a part of the International Genetically Engineered Machine Competition, or iGEM. So iGEM is a world-renowned synthetic biology competition that's hosted each year by MIT, and students at the high school, undergraduate, and overgraduate levels work from January to November on a particular project within the realm of synthetic biology and genetic engineering to solve problems and provide solutions to issues on all scales. So the work that was carried out here was actually completed by 2 iGEM teams, and these are our 2020 team, or phase 1, and our 2021 or phase 2 teams. So as this was a project that was completed over 2 years and was not limited to just a scientific pursuit, much of what we accomplished cannot be discussed here today. But you can learn a lot more about everything that we've done on our websites, or more commonly known as our wikis, and I'll be sharing a link to those at the end of the presentation. So the origins of our 2020 and 2021 projects can actually be traced back to 2019. So after participating in this competition, I was quite literally left awestruck by everything that I'd witnessed, by everything that I'd seen my peers be able to achieve. And I was particularly inspired by the use of protein-based materials from rather unlikely sources in tissue engineering applications. And this was the case of iGEM Leiden. So they used a succerin protein derived from the Humboldt squid to develop an antimicrobial hydrogel for burn wound usage. And I was left asking myself, how can I be a part of this movement? How can we as a team at KCL advance regenerative medicine and tissue engineering with biomaterials and synthetic biology? And I was particularly interested in neuroregeneration because I really quite like neuroscience, and I feel this is rather the holy grail of regenerative medicine. And I specifically wanted to focus on spinal cord injury, as I'd been doing quite a bit of research into this topic, and I felt there was rather a significant need for clinical innovation here. And so this became the problem that we were looking to solve. But in order to solve it, we have to understand it in quite depth. So just briefly, spinal cord injury is caused by trauma to the spinal cord, which can be in the form of a compression, laceration, or contusion. And this results in net damage to both the neural and the vascular system. So the rather severe severing of the axons, mast cell death, and subsequent formation of the cyst and the glial scar disrupt the flow of information between the brain and the rest of the body. And this leads to a partial or complete loss of sensation below the site of injury. So paraplegia, tetraplegia, quadriplegia are all potential clinical outcomes of spinal cord injury. And these can have rather severe physical, mental, and economic effects on both the individual and societal level. So after we conducted a rather thorough literature review into the pathophysiology of spinal cord and its current treatments, we found that the main way in which biomaterials can be used as a treatment is as a scaffolding material that can bridge this gap created by the spinal cord and can allow for like tissue continuity and encourage axonal regrowth. So we decided to develop a novel scaffolding system or a novel nerve guidance channel that was biocompatible, biodegradable, and inexpensive, and focused on functional restoration rather than just management of the condition. So in summary, our project focused on the design and modeling of a novel polycaprolactone scaffold that was encoded in a protein-based adhesive. So polycaprolactone is a rather good polymer for biomedical applications. It's inert and it has a good degradation profile, but one of the very significant limitations of it is its lack of adhesiveness. So we decided to increase the adhesiveness of this surface by incorporating a biocompatible adhesive coating. And in order to develop, figure out what sort of source material for this adhesive, we, much like iGEM Leiden, we looked to the sea and we came across these muscle foot proteins, specifically PVFP5. So PVFP5 can be polymerized and then cross-linked and then can form this adhesive polymer that can form a monolayer along the surface of the scaffold. And this is a very adhesive material. So the scaffold itself can be 3D printed and can also fit the dimensions of the patient's glial scar. So the glial scar is pretty much removed and then the system is implanted. Additionally, it contains this open path without core macro architecture and these appropriate pores of a specific size that can encourage neural cell growth and attachment. So the overall aim of this approach was to encourage neural attachment and bidirectional growth along the surface of the scaffold to reconnect the severed axons and restore functionality. So in summary, our project can be divided into two core research groups. And this is the muscle foot protein and the 3D printing scaffold research. So why use PVFP5 as an adhesive polymer? So PVFP5 is a muscle foot protein that's derived from Paravinus viridis. This is a muscle species, and it was chosen as our source of biopolymer because it's a very adhesive protein and it has been shown to be biocompatible in the past. So muscle foot proteins themselves are a group of organic polymers that are produced by muscles and allow them to adhere to surfaces, particularly those in aqueous and rather turbulent environments. So PVFP5 is found at the interface between the surface and the bias. So it has a particularly good at displacing water molecules and it's particularly adhesive. And the adhesiveness of these proteins is due to the presence of these tyrosine residues, which undergo hydroxylation and oxidation, as we can see here in this schematic. And this forms L-dopa and dopaquinone, which are responsible for the adhesiveness and cohesiveness of the polymeric material. And this reaction is catalyzed by a tyrosinase enzyme. So in summary, we decided to exploit these intrinsic properties of the protein polymer network to create this bioadhesive material. And to do so, we carried out the following practical work. So firstly, we were able to achieve recombinant PVFP5 expression in a variety of E. coli cell lines, particularly those which are specialized for disulfide bridge formation. And we co-express this with the tyrosinase enzyme, which is used to catalyze the reaction I showed previously. And in particular, we use BAMTIR, which is an enzyme secreted by Bacillus megaterium. So although we were able to achieve this expression, we had quite a number of issues with purification and isolating the protein, particularly in high quantities, due to its intrinsic adhesiveness and tendency to form aggregates. So with regards to our dry lab work, we provided the first structural model of this protein in terms computationally, using molecular dynamic simulations in GROMACS. And we also, in the phase 2 element of our work, modeled the binding of PBFP5 to the PCL surface. And if you would like to learn more about this, we do have a video on our YouTube channel explaining it, and I would definitely recommend checking it out. So with regards, with regards to our 3D bioprintable scaffold, so in the phase 2 element of our work, we designed and modeled the 3D bioprintable polycaprylicatone scaffold, and we ensured that it could be tailored to the patient. So it can be printed with the dimensions of the patient using MIR scans, and these can be segmented in ITK snap, and then these can be used to create CAD images that we can incorporate into our specific microarchitecture design so they can have the appropriate patient dimensions. And the second phase of our work built upon everything that we had done in our first year. So after we realized that this initial design was too complex to create the functional mesh or like the pores that we had desired, a new log pile design was generated, and this still allowed us to maintain our high pore connectivity and the optimal pore size, but it required less computational work and effort for the printing process. So just briefly, in our first year, we had created an algorithm that could design the pores of a particular size, but it was a very computationally heavy endeavor. So our team also applied a number of modeling approaches, including finite element analysis of the macro architecture and scaffold degradation to investigate how our scaffold will degrade alongside axonal regrowth and to ensure that it would remain mechanically sound throughout the entire regeneration stage. So in the second phase, we also carried out computational fluid dynamic simulations so that we could investigate the effect of cerebral spinal fluid on our design. And this allowed us to actually modify our macro architecture. So a third and more recent element of our project is the use of injectable Crondot today's ABC. So this is a bacterial enzyme that has been shown to encourage plasticity and axonal regeneration in the central nervous system. So as a part of our design, we have incorporated the use of microinjections during the scaffold implantation and after scaffold implantation, the timing of which is determined by imaging, so we can see the axonal regrowth scores in this manner. So these microinjections have been shown to increase the permissiveness of the spinal cord injury environment, such as a glial scar for axonal regeneration. However, it is not a stable protein for long periods of time at body temperature. So we decided to use protein engineering techniques to make this more thermostable. In particular, we introduced 7 new mutations to the sequence in order to make it into a more thermostable protein, and we were able to do so successfully. So in conclusion, our project is quite foundational and requires an abundance of further research to be taken even remotely towards the clinical sphere. Despite this, we are quite proud of everything that we achieved. In 2020, we received a gold medal and a trophy for Best Therapeutics Project in the overgraduate division. In 2021, we received a gold medal as well and a Best Presentation Award in the undergraduate division. Those trophies should be in New Hunt's house sometime soon, so if you have a chance, go check those out. And we're hoping that others will be inspired by our work and will continue to progress regenerative solutions for spinal cord injury using our approach. And that brings me to the end of my presentation. Thank you so much for listening. And I'd like to express thanks to our sponsors, which are shown here, and also a huge thank you to our PIs, Dr. Anatoly Markiv and Dr. Alison Snape, and our advisors, Professor Annalisa Pastore and Dr. Katerina Alfano. As I've mentioned, we have documented everything quite thoroughly on our wikis, and the links to which can be found here. So if you'd like to just read more on what we've done. And I guess this is me, and if you have any questions at all about iGEM or about our research, do not hesitate to reach out here by email or LinkedIn. Thank you.
Thank you very much, Abigail. This was really inspiring talk. And it is really interesting to see, both you and Ed previously, how you got inspiration by looking into kind of lower level organisms, in his case, algae, and in your case, going into mussels, which, usually we see on our plates, usually kind of in regular situations and don't really think about adhesive properties, but this was an excellent idea. We have several questions here. I will not be able to ask all of them in the interest of time, but how have you actually tested how this new scaffold, which just kind of in a way your idea is to keep together transected parts of the spinal cord. How does it affect the formation of the glial scar, which of course is one of the biggest problems, and that's why you use chondroitinase ABC?
So it does have the potential, because the overall surgical procedure would be to remove the glial scar and then implant the material in. And naturally, like after this surgical procedure, there's potential to lead to further glial scar formation. So it has the potential to, but these would be much smaller than the initial lesion that would be removed. So it does, in summary, have the potential to cause a further glial scar formation, but it would be on a much smaller scale. And that's why we have incorporated the CHABC enzyme to accommodate for that.
So, if I understand you correctly, you envisage that this type of scaffolding will be actually also very useful, not just for acutely injured patients, but also those with chronic injury, right?
Yes, actually, more of our design was tailored towards the chronic injury, primarily because of the rather extensive inflammation and immune response that happens in the acute phase, and just the plethora of cellular events that are taking place. So we initially anticipated waiting until it stabilizes, and then actually once the glial scar has formed, then actually implement the surgery. And that's yeah, that was our therapeutic approach.
Okay, so two more questions. One is, have you actually already run some preclinical testing on mice and rats? That was the question from the audience.
No, we haven't. So we have 3D printed the scaffold. As I mentioned, we did this as a part of iGEM competition, and there's quite significant restrictions on what you can actually do within the competition. So we aren't really allowed to work with higher order organisms. So that's something that if we would like to see some other students, like people who have taken part in the project, take this to the next step, I would definitely be interested in scaling it up and actually pursuing it in that regard. But it's something that we haven't achieved yet.
Oh, but actually wonderful. I think that there is a great potential for kind of collaborating with great researchers in the field of spinal cord injury, Liz Bradbury here at King's, Martin Schwab in Switzerland, and so on. And when I'm mentioning Martin Schwab, actually, have you been thinking maybe of using not just kind of anti-glial, let's call them like that, enzymes, like chondroitinase ABC, but maybe using something like anti-NOGA antibodies to basically unblock the blockers, remove the blockers blocking the axonal regeneration?
Isn't something that we explored in much depth. I think that's something that we touched upon very briefly in our first phase. We very briefly looked at other small molecule or drug delivery approaches using the scaffold, but it's something that we didn't explore in depth because it complicated our design. And for just a year, it was a bit much. But it's definitely something that we could potentially look into. And Liz Bradbury is who suggested for us to use CHABC. So we did work with her quite a great deal. So something that's very important to the iGEM competition is this idea of human practices. And it's basically to ensure that your project only benefits the world in a positive way. And that requires very extensive interaction with our stakeholders, like academic, clinical, and industrial. So we spoke with a lot of people at King's and abroad to gain a lot of advice as to how to best adapt our project. And we also had the huge privilege to speak with the Spinal Cord Injury Association as well. So yeah, we did take into account a lot of different different perspectives throughout the design.
Excellent. Thank you very much for this excellent presentation, and good luck with the future development of this project. And now we are moving to our last talk for this conference, and this is going to be a keynote talk delivered by Professor Dustin Tyler. Dustin is a Kent H. Smith Professor of Biomedical Engineering at Case Western Reserve University. He's also Director of the Human Fusions Institute, Associate Director of Advanced Platform Technologies Center at the Cleveland Veterans Affairs Medical Center. And Dustin will tell us something about human technology symbiosis with neurotechnologies. Dustin, thank you very much for joining us today, and we are looking forward to hear your talk now.
Thanks, Maria. Thanks, everybody, for the invite to be here. It's kind of humbling to follow such brilliant talks, but I appreciate the time to talk about the work we're doing. And actually really, hopefully, engender a discussion later about the human-technology relationship. And so this talk will be a bit more, we started at a very cellular, molecular, protein level with Ed's talk and the awesome work he's doing and moving up. This I want to talk about sort of thinking about how do humans and technology co-evolve. And if you think about this, humans are probably the only species, if you will, that really relies on technology. We're exceptionally intertwined. I live here in Cleveland. We're heading into winter. I'm sure in northern England and in different places. If I don't have clothes, if I don't have a building, and I'm outside for an evening, I'm probably done, right? I rely on technology. My desk in front of me has an iPad. It has a computer. It has 2 monitors. I have digital paper. We're intertwined hopelessly with technology. I mean, we really have been since the early days of picking up stones and rocks and wearing furs. The question is, how are we co-evolving and what does the future potentially look like? And I would contend or want to talk about the idea that up to this point, we've really used technology. So we'll touch it with our hands, we type on it, we see it, we hear it. But I think we're on the cusp of moving forward because of directly connecting to the nervous system. This is what I'm going to talk about through this session. We're at the sort of transition of becoming embodied with that technology, where technology becomes an extension of ourselves in a really unique and interesting way. And that's the content of what I want to get at here. And in that regard, what we're looking at is, what is the human potential, right? I want to put this in the concept of how do we get better? So what is the human potential? How do we scale the human potential? And what are our limitations? And if we think about this, or the way I think about it anyway, is really the intellectual capability of the brain, the sort of unique thing that we call conscious. And I'm not really here to discuss, although I find it fascinating on the panel, it'll be interesting to think about what conscious is specifically, but the capabilities of the human mind and what we do are huge. We're still trying to create the artificial intelligence, which I think is a misnomer, but machine learning is trying to understand how to use sort of the same concepts of the human brain. But that is a potential. What are our limitations? Essentially, we are limited currently by our skin in many ways, right? The extent of where I reach to, the extent of my boundaries are bound by the sensory input that I have. It's where I physically create a bound around me. I'm also limited by the biology I have. The muscles have a certain amount of power and capability, them certain strengths that I as an individual can do. And I'm not very resilient. There's a lot of environments in which I'm going to not succeed. So there's a lot of potential. How can we scale that up? How can we expand it using this integration with technology? And so that's the essence of the talk. To start, I'm going to kind of make the human simple, right? So what is the human? This is a concept that kind of came out around the 1950s. But if you really think about what we are, we're a message processor. And so I can kind of, the very simple view I start with some of my classes of what the human is you have an input to your system, which essentially is several energy forms: mechanical, light, chemical, temperature. Those are the human forms. Of course, other species have magnetic or different fields, but this is really the input we have to our different senses. That's how we experience the world. That's communicated by peripheral nerves, different nervous systems to the brain, the central system processing, right? That takes an input message, assembles all that, puts all of our experience that it's had from our time of birth all the way through, makes a decision, makes some output, and pushes that back through that communication pathway, the peripheral nerves, and out into the output, which effectively to the environment are our muscles and glands and organs. Those are pretty much the output of our system. And so in a very simple level, we can just think of this as one continuous loop where we're bringing in information, we're processing it in a very complex and an amazing system, which you heard a lot about different ways of learning about it and what it's doing. There's far more in those areas. And then we push that back out to an activity. When I'm speaking, it's a very coordinated activity of muscles to produce sound, to produce environment, to go back to that input system. And so on a very crude level, we can look at the human as this system, and I'm going to talk about that in various ways. So the neural interface, the idea that my background and where we're pushing is the neural interface is kind of bypassing that input system and that output system by being able to directly communicate to this message processor, both on the input side, which is what I'll spend most of my time on in this talk, and also at the output side, where I can either directly intervene by stimulating the pathways to cause output, or I can get information from that message processor to interpret what that message processing was. On the input side, I can stimulate and actually provide information independent of the input system, and you'll see this come through here in the rest of the slides. And what is that nervous system? It's just on a very gross level. You've got the central nervous system, which is everything in the spinal cord and the brain, right? Or in the skull, your brain and your spinal cord. Peripheral nervous system, which is all to the extremities, and then versions of that, which are the sympathetic and parasympathetic systems, the autonomic system, which is internal regulation. And so these are all connected by this incredible network. And then, as you've heard about earlier, and we know, is the brain, the processor part of this. So there are a lot of different interfaces. This is just kind of a survey to get you an idea. I'm going to talk mainly about peripheral interfaces of different ways of communicating to those nerves that are in the periphery, and I'll explain why I find that particularly exciting. There's aware, and this is just an example of a few, like micro-ECOG arrays, intracortical arrays, there's ECOG arrays, there's all kinds of different ways of interacting with that nervous system, and they're continuing to grow every day. So we're learning how to communicate with that system, and that field of technology is growing very rapidly. Where has it been used? These are things that are going on in the Cleveland area and around. We heard about spinal cord injury and repairing the spinal cord, but for the cases are still, while it's not possible, we're stimulating directly with the motor system for people with quadriplegia, where we can stimulate the muscle activity to cause motion. We've been doing this for over 10 years with subjects with direct peripheral nerve interfaces in quadriplegia, in paraplegia, even dysphagia for swallowing management following stroke. So this is where we're stimulating the system to cause output and movement of the human. We can move beyond that then in sort of the autonomic system. We're working at, and we and others are working at ways of stimulating, for example, nerves in the neck that reflect the blood pressure, and how we can control hypertension. So controlling that system, right? This whole idea of biopharmaceuticals, bioelectronic medicine is how do we use the body system to maintain its own homeostasis, and that things like epilepsy, rheumatoid arthritis, depression, even in memory states have all been areas of stimulating the peripheral nerve to affect the way that the body operates. On the sensory side, which is I'm going to spend most of the time talking about our work and then extend that, we're working now in areas like upper limb loss, lower limb loss, and vascular disease to now put information into that sensory system to work with the brain. Things like movement disorders, psychology also have areas where I think that these input systems, sensory feedback, can help with movement disorder, can help with psychological elements in in-depth virtual reality environments. And I'm not going to talk about that, but there's a lot of fascinating work going on in that environment. So that's just a broad survey. What I want to talk about specifically and get to, for examples of myself, and then expand this idea, starts with work that we were doing in prosthetic interfaces. So when somebody loses a limb, when I started about 10 years ago, 15 years ago, kind of the ignorant engineer, I was very interested in the mechanics of the hand and how we create an interesting hand. But as a little more enlightened, but not enlightened, I'm sure. But as I've been talking with our subjects over time, what I find is that it's not about the function of the hand. We ask every one of them, every potential candidate, what do you want? And none of them has ever said that I want to use my hand better. Every single person I've ever talked to, the first thing they say is I want to hold my wife's hand. I want to talk, I want to connect with my children. And so it really started to rethink what the point of sensory interface and connection to the world is all about. Let me give you a bit of an example of where we are. And so this is sort of the kind of the leading edge of the technology that exists in terms of clinical implementation, particularly prosthetics. This is work we just completed after about, about 5 years now, with partners like Medtronic, Ardea Medical, Lawrence Livermore Lab, and various others in the prosthetics area. We now have created this complete system where it's fully implanted. So there's a device set in the chest that's connected to the outside world by Bluetooth, powered several days. It's rechargeable battery, connects to several leads in the body that can do up to 64 stimulation channels for sensory input. We can record from 32 different muscles. So that's the input system on that diagram I showed you. We can connect to that. Then we can record from the output system I showed you. all Bluetooth connected, and we currently are working under FDA approval and an IDE trial to do an initial randomized clinical trial of this device here in the US. And in fact, we just implanted 2 weeks ago our first system. This is half of it laying here that you can see. Tomorrow that subject comes back to get the system turned on in sensory for the first time. So very exciting. Before this system, which is what's indicated here, we just started, we've actually had people for up to 9 years now with without the full implant, but with the neural interface technology that have been getting sensory input and then connecting to a prosthetic hand. So what we get is when we stimulate the nerves, they don't say, in fact, one of the first times the subject came on, the first time we turned on current about 9 years ago, for a subject kind of, you know, he's been without his hand for about 2 years. As soon as we turn the current on, he kind of got this funny look in his face. He stops for a second. He says, you know, that's my thumb. I haven't felt my thumb since the accident. Now he has a phantom, but he said, that's my thumb. And it's the first time I felt it since the accident. So by stimulating the nerve directly, we bypass the sensors that are lost, but we're still introducing into the communication system that existed. And the brain interprets it as though it came from the world around it. That is his thumb. And we've been able to do this now we have 4 subjects over a period of 9 years. We've been able to, more recently, about 5 years ago, we started recording. So when the person thinks about moving their hand, which no longer exists, we record the muscle activity that would have controlled the fingers, use machine learning techniques, and actually control the hand as though they meant to control their hand. So it becomes a very natural activity. So they control the hand, they touch something, they feel it, it becomes entirely their hand. We're also doing this in lower extremity. We have now for the last 5 years as well, 4 subjects in the lower extremity. So what we've been able to do over this time, and I'm not going to go into the details for the duration, you know, for the essence of this talk is very high level, and I want to get to a bigger idea here that this leads us to. But over this time, we've had a really great tool to directly study in the human what the impacts of sensory stimulation of the input to that message processor does. Early on, basically, we showed that we can reduce the fundamentals of touch, or we can reproduce like what it feels like, where it feels like, how much pressure, all the fundamentals of touch. We can affect phantom pain. We can give proprioception and sense of movement. We've been able to control high degree of freedom prostheses. This device has been at home use, and like I mentioned earlier, connecting to people, it's changed their quality of life. They actually feel more connected around them. They can produce, or they can do more work in their home. But what's more interesting is of late, and kind of where we're heading, is it's a really more detailed work about how the brain processes information. And you know, there's several talks that could be given on each of these, so this is just a wet the whistle type of thing. But when we started this work, we thought that the mapping that existed to what was your index finger and what's the line here that went to your brain is pretty fixed. I have to stimulate it appropriately for me to be able to feel my finger. Well, it turns out, we all know the brain is very plastic. It's incredibly plastic. And in fact, the sense of the world is a multisensory integration. When you touch something, what you're seeing affects what you feel. So even if you stimulate the exact same fibers, but give a different visual input, the person will report a different output, a different expectation. And you can think about this from the perspective of you only have one experience of the world. When you see the world around you, your brain is taking in, your message processor is taking in things from vision, from touch, from smell, and it creates one representation of the world. If those sensory inputs are out of whack, and the visual illusions are a classic example of this, if they don't totally match, it's a weighted sum of that experience. And so multisensory integration becomes quite fascinating as to how we take information from all these different elements of the world and create our own reality, our perception of what exists. We know we can learn that, so we can actually start putting information into the peripheral system that's learned in different ways. So your perceptual learning about how you interact with that world. Pre-perception engagement, in other words, what I mean from that, another set of talks or another ideas is that people often talk about, you know, the literature talks a lot about sensory input to feel your hand. However, that's only a very small part of what sensory is. That's the perception of the world. Pre-perceptual engagement actually is where things are much more interesting. Your brain is far more complex than just what you feel, right? It's the emotional states and the memory. It's all those other things that happen that create you. They're not just that you feel your finger. For example, if you think of smelling chocolate chip cookies, for me, that reminds me of my grandmother. And my grandmother brings with it a whole bunch of other things that are integrations of sensory experiences from being my grandmother. Those are all integrations of who we are. It's our conception of reality. And that's the sensory input, is pre-perception engagement. Motor planning, how our system, I don't think about everything I do when I move my hand. I only think I want to move the hand. Everything else is occurring pre-perception. And so do we engage those appropriately? And that's related to sensory motor integration. So we have to get over this idea that sensory input is about what I feel. Sensory input is about how it integrates with the brain. And this is what's interesting to me when you think about BCI and brain-computer interfaces. People are really enamored with the brain component of that. But I think I want to point out and I want to emphasize that brain-computer interfaces is a misnomer. You don't interface with the brain, you interface with a very small element of the brain, often cortical interfaces, right? So DBS, it's a very small region down in the thalamus or in the basal ganglia. It's a small region of the cortex, which is perception, but the brain is much more complex. And while you take like Kandel and Schwartz, which is a classic book that shows touch as a single pathway up to the perception of the cortex, it's actually far more interesting. Even in the cuneate nucleus, the first place that you have a fundamental relay station, it breaks out into many different areas of the brain for low-level processing coordination. And this is not meant to be understood other than it's a very complex. So a touch here is not just what you feel in the cortex, but it's what goes throughout the entire brain. You're processing your entire you in many ways, right? Just to put a very simplified version of that. And so for me, the idea here is a cortical or brain interface is a limited version at the end of this chain or in the middle of it, where if I can interact from the peripheral on the input, I actually am engaging all of this information as we go. And that's where our research is at right now and things we're looking at, how does that affect it? But here's where I want to go for the last couple minutes I have in this talk now. So that gives you the sense that we're able to talk to that message processor in the absence of the physical input. And so some of the examples here that have been influential of our subjects kind of comments to us. This is one as far as the connection piece. This is a first picture one of our subjects sent to us, and we've used this a lot, so he's fine with this in public domain. He sent it and he said, before I had the system, without it, I was only able to pick up my granddaughter with my remaining arm. I would never use the prosthesis because I didn't know if I was squeezing too hard. But with the sensation, with this new system, I picked her right up. She came over, I just grabbed her and picked her up because I could feel her. And so that was really impactful to him, but also to us in the concept of what we're doing in this connection to people. And then this is where I want to jump to the next big thing here to finish out, is that our subjects say with the prosthesis, without sensation, it's just a tool at the end of the arm. It's like you or I using a hammer or a screwdriver. But as soon as we turn the system on, we connect to that sensory system, it's their hand. They actually start talking about them grabbing things with their hand, not with the prosthesis. Here's where it gets really interesting, is that now this sensation, this skin, their sensor, is purely a digital stream of data that we've created from an engineering perspective, and I'm applying it on that that system, right, the communication piece. I can put this prosthesis anywhere in the world and it will still feel exactly the same. It doesn't matter that it's connected to them. So now I can essentially put their hand in London, right? And you can now connect this data over the web. And now imagine if he was grabbing something in London, he would feel exactly the same thing as if he's feeling it in his prosthesis in Cleveland. And so this gets to, now let's complicate this message processor idea, and more, I think what the future holds is that we have the input system. This is what's on our skin and our current abilities. However, virtual reality, so we all know about virtual reality is big now, metaverse, and we aren't getting into the discussion right now of what you think of all that, but virtual reality connects with the input systems that exist. We're working on this idea of neural reality where I can bypass that. I can go directly to the nervous system and connect to that message processor. And now that system connects to remote and virtual environments over the network as though it's their own. And so, and this is the case of neural reality is bypassing both our input sensors. So I can put any data in here. I could put magnetic field data onto the tactile stream if I want. I can give you magnetic sensation if so desired. We can make ultrasound in medical reality feel like grabbing something. So we can totally change the way that the human engages with the world. What that allows us to do, or what the way we're looking at this is the human fusions idea, is we can take this human capability, that message processor, right, as a gross term, our intellectual capability, our creativity, the lifelong experiences we have, add that to technology where now we can say, we can put in the networking, the robotics, the computational, machine learning, the pieces where we are weaker, combine them to get better, to scale that human, to kind of get this transforming the human technology relationship for a thriving, just, and connected society. So now we can think about scaling the human because we're no longer limited by our skin. So now I can connect you over the neural system at anywhere, anytime. the capability is limited by what I connect you to. So no longer the biologic system, but what you're connected into. And of course, we're resilient because I can put things in the bottom of the ocean and you can operate there. So it's quite fascinating what we can do. Essentially, we're working now with systems where we use AR, VR for the video, audio, because they're well advanced, but the tactile components, putting together on the nervous system over this idea of neural. So I talked about prosthetics, it's implanted. Nobody's going to be doing that right now, maybe someday. We're working with wearables now. We're stimulating the nerves directly, not the hand, and we can remove some of the bulk of current VR devices. And then minimally invasive, where someday maybe we can do this just by a shot. And so now you're directly connected to the nervous system. And once you're there, you have the entire world connected to you if you so desire by a Bluetooth connection or something. We're creating a platform for that to be on. We have networks now set up. We've actually been communicating regularly, controlling a robot in UCLA from a person in Cleveland. And this is an example of such. This is a robot that was developed out at UCLA. What this person's wearing is the VR set. This glove is while we stimulate right now with the peripheral nervous system. And so he's been controlling while he's sitting there with it together. He's been able to control this robot across, basically across the country, the US anyway, and from Cleveland. And so we did this in a judge's competition. She's doing it after 15 minutes of training. We've been working with colleagues there. So this, we think, has applications in healthcare, space, industry, military, entertainment. You can start to think about all kinds of things I would love to talk about. Time won't permit that. And this is where I want to end on, right? So this is what, when you start to think about this, and maybe I watched too much of the Black Mirror, which I think is a British, a fantastic show, a little dystopian of technology, but it's a great show, at least the first couple seasons. But to do this, a lot of technology often starts and ends right here, where tech develops tech because it wants to. And what I think is the beauty of this conference, and what we really need to do much more, and I'm super interested in, is we can ask the technical questions of how do we build it. We're very good about that. And technology does that. It's why it's exploding so rapidly. And we can put it in the clinical, we can put it in the people. But really, the question isn't so much of how do we build it. We should also put around that, should we do it? What are the boundaries? Technology itself is neither good nor bad. But if we think about the ethical ways of going forward, we can actually do it in the right way and solve the problem before it starts, right? So if we develop something that goes to 1 billion people and say, hey, maybe we should think about the ethics, it's just too late. How do we get it from the beginning? And then once we know what the ethical sides are, what's the regulatory world? How do we manage it so it stays in an ethical environment as we translate it out to make sure it gets to the community, which is very important? And then how does it change the society? How does society and technology co-evolve, which leads back to this loop? And this is sort of what we look at as the broad collaboration that we're trying to develop in the human fusions, but also what I think we need to be going forward. And so what I'll end you with, maybe to seed the panel a little bit, from my thoughts anyway, about sort of when we start thinking about the human technology symbiosis and where we're going, is now you start to think about interesting things, that the skin is no longer our boundary. If I can connect you to digital systems that are your experience or maybe other experiences, who owns your experience? Who owns your expertise? If you're doing things and I'm controlling something in California, all that data that's going between there is available potentially. Now you're getting my capability as I'm doing something, the intelligence, now that message processor is in the loop and you're connecting the input output, as that's being learned, who owns that? What is the boundary between individuals? That's no longer the skin. I can connect in different ways that we never could before. I could, for example, in texting, you text a message, but I could actually text you a feeling. We could text a handshake. So now where are the boundaries and what are the limits to that shared experience? Where do we put boundaries and controls on that? How do we include all voices? I think this is a classic thing. How do we not widen the digital divide and the gender and the URM gaps, but how do we include that? So rather than a small group of people developing technology for themselves, how do we develop for everyone? How do we democratize this technology? How do we make it available? And how do we use it to create a just society? How are the experiences of neural reality and the activity you do protected for privacy, and intrusion? If I'm out on the web and somebody puts a stream in there, how do we protect that? And then you can actually get to, and this is something I would love to talk about, maybe this, beyond this, but what is reality becomes a really interesting question. And I'll stop there. This is kind of a lot of people have been working on this. It's way too many to mention, but this will be online. You can take a look later. This is on the human fusion side, an awesome group of people have been working with. And I will stop there at this point for any questions you might have here. And I look forward to hopefully a vigorous panel discussion. So thank you for your time.
Dustin, thank you for this talk. I'm actually at the moment quite speechless. Abilities that we have at this time and age, technology that we have is fascinating, but as you said, it can be very scary. And I would actually like to stop here probably and make a short break so that we can really start digesting all of these things before we move into a panel. Because you have opened so many questions now that if we now start with questions, we will just, there won't be any break. We'll just continue, you know, for hours like this. But you have raised something that we have mentioned earlier, and that I believe, you know, started probably a long time ago with, you know, during the World War II with Project Manhattan. We have the technology, but do we really need to use it? This is a different scale now.
Yeah.
And
well,
I think, yeah.
Sorry, I don't mean to interrupt you, but I just want to kind of leave a thought with you, because people start to get, I want to shape this conversation maybe a little bit, because people kind of go, wow, this is so different, but that has happened at every technological revolution throughout the way, right? People have always been engaged with technology. At one point when television came out, it was going to destroy the world, right? So I think that I hope, and what I want to sort of say before we go to the panel is, it's not like, oh my God, this is going to change the world, this is super scary. It's rather, you know, we've been doing this all along. Let's think about it from a more rational perspective and actually say it's not itself scary, but let's really think rationally about what it is. You know, for example, radio is not scary, but it's what we're doing here. It's taking one form of energy I can't possibly perceive with my own system, and I put it through a box that augments me because it gives me that into an audio version. What we're doing by Zoom. So it's not, it's interesting to a new direction, but it's not, I hope to not do it by fear. We just have to be rational. And I think that's the key thing. So I'm sorry to interrupt, but I just want to make sure we kind of scope it around that.
I agree with you. As you said very nicely, you know, nothing per se, including technology, is good or bad. It's what we do with that. Mm-hmm. So I'm really looking forward to this panel. There will be lots of discussion, and I really encourage our audience to join us. Post the questions in the forum. All the questions are anonymous. So, you know, if you wonder that you're asking maybe something controversial or something, and somebody will know who you are, no, we won't. So please do post your questions, and let's be back in 5 minutes. So just grab your new mug of coffee or tea, wherever you are. If you're in UK, then definitely tea. This is a tea time almost. And let's be back in 5 minutes to 3 p.m. London time, and let's have a great discussion with Dustin, Jack, and Charlie about ethics of neurotechnologies. We are ready to continue. Welcome back after a short break. I hope that you got something to eat and got your questions sorted out. Definitely not enough time to think about all the questions that we can think about after hearing all of those talks in the last these 2 days. So if you think what we have heard since yesterday, there was really a lot. We have started yesterday talking about artificial intelligence, virtual reality. We have also explored uses of TMS and DBS. Then we moved on to iPSC, so more cellular methodologies. We have heard today about optogenetics, Anchor 1, looking into computation of a cell, you know, in a real time from different angles. We have heard about new strategies, how to deal with spinal cord injuries and limb loss, and so on and so on. So all of these methodologies raise so many questions, and as Dustin actually said at one point, technologically we are incredibly advanced. We can build almost anything. But legislation and ethics usually these days lag behind, because as we have seen, especially at the first talk today, just in the last 15 years, before the last 15 years, kind of technological developments were fast, but probably not as fast as during the last decade and a half or 2 decades, when we have something new coming almost every month. And of course, that legislation, ethics, and everything can't really keep up because politics is usually much slower than you know, our technological developments. So how can we deal with this? How can we use technology, like for example, DBS and TMS, which are in use for many years, but we still don't know exact mechanisms how they work. They work like many pharmacological treatments. Paracetamol, for example, we know it works, but we still don't know exact mechanism. So I would really like to explore all of these issues and all of these topics with our speakers, Professor Dustin Tyler, Professor Jack Price, and Dr. Charlie Winford-Smith, to see where are we going, how we deal with all of these things. Is everything is ethical to do whatever we can, or we need to put brakes somewhere? And when we have a patient who needs help, can we just go in, or we need some very, very rigorously controlled trial, and so on and so on. So Charlie, Jack, and Dustin, are you with us? And our audience, the questions that you would like to ask, please just put them all into the chat. So Dustin, you actually had this wonderful slide at the end, which you are opening, so many questions, and one of them, I believe, was about our privacy.
Mm-hmm.
Who is the person our experience? Where are the boundaries now between the people? Who is owning the experience? Can you comment on that a little bit, especially with the technologies that you are developing?
So, I mean, the thought there, right, is, Europe has a whole bunch of different privacy laws. We have all these things about our search data. What we look for is now, and think about how much of our fingerprint, right? So even the security stuff where you're going to not store here or there, you can create a fingerprint of somebody by taking data across all points of the web. It's virtually impossible not to know who you are because you're sharing yourself in some different way. What's that on the next level, right? I mean, so that's thinking about things I do on the web in terms of, just my information out there. But now we're at the point where you're connecting to you in a much different way, right? I mean, this is your experience. This is your use of things. It could be, for example, holding your kid's hand or playing with a grandkid across the country, right? So imagine a day where you could basically be not in a telepresence like we do now, right? But you could actually be through a hollow system or something of that nature, holding your grandkid's hand someplace else, right? And think of Oculus right now, everything to use the Oculus system, every piece goes to Facebook. To use Oculus, you agree that everything you're doing goes to Facebook. That's scary to me, right? I mean, essentially, yes, they're going to do it to make your experience better, but I don't buy that. Do you? And so where is that boundary? Like, what do you own? What should you have in the privacy? And then, there is value to being able to improve the system so we get better. And how do we protect that? How do you keep your experience yours? When do you own it? And then the second corollary to that question was thinking of intelligent US applications, say not US, use applications. Imagine you're an expert doing a system, right? So one of the advantages of a sort of neural reality type of telepresence or a neural presence is that you could actually be someplace else, conceivably providing your expertise, which is sort of the mental side, doing a task. So let's say you're in some industry where you have a specialized skill that when you're working with an object, you can do something other people can't because it's learned. You've learned the field, you learned how to manipulate the material to do whatever you're doing. That before was inherent. You couldn't really collect what was going on. You kind of learned it, right? But now the sensory information that you're using, right, is now digitally available before it becomes to you. Your decision-making process you do still with your expertise, and then what you do with that information now is also recordable going back. You can pull the input-output of that system, which is a classic system, identification program or idea. So now if we're using machine learning to learn your expertise, it took you forever to get that. Does it become freely available? Like, if you have a machine learning system that can now create your expertise, is that like music by somebody created? You pay for every use. I mean, how do we think about what that is? And I think these boundaries become very interesting, right? Where do you end on that? And what do you own? I can stop there. Let me say one other thing about this. You start to expand this, and this I get in trouble because it's like really cool where all this goes. But I mean, the really interesting part is thinking about rules and independence and all these things that are in the news today, in a very detrimental way for some of our health even. If the idea of your boundary is your skin and you exist on your own without connection to anybody else, that's sort of it's not really the way the world works, right? We're totally interconnected in different ways, but now we're pushing that to another level. you're actually interconnected, not because I talk to you, but you could be interconnected because you're on this data stream together. You can have your you can actually physically, perceptually exist in a co-space. It kind of blurs the boundary of your individuality depending how you set this up. And so it leads to a whole lot of really interesting thoughts about you, what you own versus society, what is society, etc. So I kind of stopped there because it starts to fall off the deep end and I don't want to go that far away, but it does ask some really interesting questions about what you own, what's your experience, and then where we go with that.
It's kind of almost like a sci-fi movie from the 70s and 80s, which is coming to the reality, kind of sharing the experience, because, you know, why we like literature, why we like art, it's the experience of it, which is very often so difficult to explain to somebody.
Yep. Well, and I mean, think of what we can do. Think about the art. I mean, actually think about what the future of the art is. Right now, art is experienced through your eyes, your ears, you know, etc. If I have a direct connection to your sensory input, I want to get this to artists' hands to create data streams that have an experience. I don't know what the hell it's going to be, right? That's the beauty of it. It's going to be different. And those people that know how to deal with it can create an entirely unique experience there. So, you know, from literature, how I was motivated by Star Wars, like many people in the prosthetics field, right? But literature has come up with this long ago. Clark in the Odyssey series, basically he described this system already. If you go all the way through his books, not just 2010 and 2001, 2010, that were the movies. If you go through the 3010 series, it was sort of described this idea of this entire alternate reality through a neural connection. So I would love to think I'm unique, but it turns out good sci-fi writers have already done this, which is the beauty of good lit. I mean, it actually is a, you know, who leads whom.
Indeed, I just wanted to say that, you know, many scientists will say, oh, sci-fi, I'm not really into it. It's kind of, but who leads whom? Exactly what you said. But let me just think about something else, which kind of, you know, it's probably an exaggeration at the moment, but more probably not for the future. If you're talking about sharing our experiences and expanding our consciousness and basically our experience, what about storing our experience after our life? You know, kind of having this idea of eternal life in a different form.
Yeah, so that's interesting, right? And I think this is where and I'd really talk to my colleagues on this and get their input. I think some of the misleading components where we go, right? So the misdirection about what that message processor is we try to equate the brain to a digital system. I think that's a mistake, right? Our memories are not bits, right? I mean, our brain doesn't have certain cells turned on and off. I think, you know, there's an interesting question, what is storing your memory, right? So there's stored experiences, we could do that. Storing you, I don't know what the hell that means, right? Because, I mean, think about, you exist, you're a state machine of an incredibly complex system. You got, you know, millions of neurons, billions of connections, and all the molecules that do them. We know now the stuff that Boyden and people are looking at, the molecular level of the synapse affects who you are. So how far do you have to store that to do what you just said? And I'm not sure that that's feasible, right? So and even if you put, what are you going to load it back into, right? You need exactly the same system to reload all that. So I think we have to be careful about extending this too far. There are things that we can do and things that we can learn that are important. And I think there's elements, like you could store a family vacation. So take your video camera, but add touch to it. So now you've got a stream that's not just the video, but you've also got the tactile, and you can play back that. Okay, those things we can start to do, but to record your memory for your entire life existence is still something I'm unconvinced about. So it's an interesting boundary.
Yep. Well, but very interesting topic, and I'm very happy to see that Professor Michael Nitsch join us for the panel. Michael, thank you very much for joining. We were just discussing, you know, expanding human experiences and consciousness and cognition by the interesting work and new technologies that we have explored during these last 2 days. And just continuing to this discussion with Dustin, what you have said, you know, at the beginning of your talk, you said something like, you know, we really want to, as humans, we always want to improve ourselves, improve our consciousness, improve our cognition. And this was something that Charlie also mentioned yesterday, that there are several companies in UK, US, and so on interested in that, especially improving our cognitive capacities. And of course that some agencies ministries of defense, military use, of course, everything, all of these things are interested also for those purposes. So Charlie, what is your view on all of those technologies and ethics of their use and the interest that is there for them?
I think there's definitely interest also in do-it-yourself communities and so on. So that's in sports, for example, and these things. For TDCS, WADA says it's not doping because it's a bit like if you train in high altitude to improve your performance, it won't work on its own, but it needs training. I think it's a clever move, because how will you check? How will you find out that somebody put low electricity to the brain at the end, right? So there's not really a method like in pharmacology. So there's definitely interest. As you said, two parts. One is scientifically, one is the ethics part. Scientifically, I think, and this is valid for most of the brain stimulation protocols, non-invasive brain stimulation protocols, we are able to modify performance in both directions. But as far as what I can say for TDCS, the studies which have done these were proof of principle pilot studies. And if you want to make this worthwhile if you don't can talk about clinical applications, that's one thing, and the other thing is neuroenhancement. And if you talk about neuroenhancement, as with clinical applications, you need protocols which give you optimal effects, which was not the aim and not the design of these proof of principle studies. So you need another type of studies if you want to do this. And then the question is, do you get effects which are relevant in everyday life? And this is so far, I think, a rather open question for neuroenhancement in, let's say, healthy people, healthy young people, or elderly people. Scientifically, I think there's a large motivation also to improve performance, to antagonize performance decline in higher age, which might be different to healthy young people. And the next thing is that optimization might also mean adaptation on a specific task, because we saw, for example, for motor learning, in specific tasks, and for early motor learning, improving effects with anodol TDCS, so LTP enhancement in the overlearned state. In the same task, we improve performance with cathode TDCS, most likely via signal-to-noise enhancement. And so it's not quite trivial. Just put the electrodes on, what many of the companies now do, and then everything gets better. That's more tricky, and I think it's technologically more demanding at the end. Might work in some cases, might not work in other cases. What we also don't know is, which were relevant for application and which might be critical, especially for neuroenhancement, is what happens if you don't do only one session or two sessions, but they would want to use it every day or whatever. They want to have stable effect for longer times. And then there's also the question of safety. Does it work all in the same direction, or do you get also, is it also always beneficial, or do effects get in the other direction? So for acute performance, it seems to be very safe. We didn't have serious adverse effects now for tens of thousands of people. But if at all, then prolonged stimulation has been done in patients. And there's a risk-benefit ratio is, of course, different. In patients, if there are small side effects or something like this, you might tolerate this if the principal effect is beneficial and is large enough and clinically relevant. If you have healthy subjects, then we won't be happy even if there are rare side effects. So this needs money and manpower, I think, to do this if one wants to do this. And second question is, of course, the ethical question. I think for patients, it's not really problematic. It's the same with all interventions, no principle different from pharmacological or so on. For neuroenhancement in healthy subjects, one question is the question of doping in athletes. If doping is not doping, BARDA says no. You can discuss this from different sides. I'm not a doping specialist. The other thing is, should we artificially enhance our performance? And it's partially, I think, discussed extremely critically. But when you think about what we do, neuroenhancement by ourselves, is everyday substances. People are smoking, people take alcohol to get them in a specific state. People are drinking coffee. And the question is there a qualitative difference? And should we then say no coffee anymore, no tea, no alcohol, no whatever, no Red Bull? Red Bull never anyhow, because we have this bad experience with our Leipzig football team. And this I don't like very much. But you know what I mean. So I think it's more grayish at the end. And for me, I don't see a qualitative difference if you want to get you in a specific state, if you drink your coffee or take some drugs or whatever, or you do electrical brain stimulations, and that's more a practical question. Yeah, so maybe I'm more moderate there, and maybe there are strong arguments which make this ethically problematic, but I think the discussion currently is a bit out of content, out of context, sometimes a bit hysteric also.
No, I agree with you completely, and kind of what you're saying is, where do we draw the line? Yeah. How we decide which kind of stimulant we want to use, or which is appropriate or acceptable. And we have another question from the audience, actually, that I would like to now pose. And the question is for Dustin, but actually for everybody. Please do all get involved. So our audience member says, your work is fascinating, and I'm excited to see where it goes. When driven my medical applications, we are looking to reduce, restore, recover, to simplify. What do you think about sharing negative experiences, such as pain?
Well, I'll just give a simple example for that, where pain is good, right? I mean, which sounds bad, but if you have a prosthetic, we have pain for a purpose, right? Pain protects us. And so one of the challenges in people that wear prosthetics on a very simple side is that they destroy them all the time, because they don't have, I mean, people will grab stuff off a grill. He'll use it as a hammer because it doesn't hurt, right? So pain it's a boundary, it's a protective mechanism that we have. Now, excruciating pain is a whole nother deal, right? I mean, that's when you're actually in damage. So I think if you use pain as it is as a protective mechanism, there's value there. But again, an ethical question, you know, you can create pain that isn't valuable, in fact, quite negative. And so that would be, you know, how do you prevent that? How do you get away from that, right? I mean, it's very easy to cause intense pain with no physical damage, and that has a lot of implications, right? Not good ones. So great question. Pain has value, and if we know where the value is, we can use it for that. But you know, we have to be careful about this. Another one of those questions. And you know, I'll say around some of this too, and I think it all wraps of this, and I'll stop. But you know, we say it's good or bad. I don't know what good or bad means. This is where the ethic is really important, right? Is it good because I define it good because I'm better? How's the societal effect of that? So what I think we need to have as a discussion as a whole is, what does good and bad mean? How do we know? And it goes with what Miko was saying, right? What is good? Like, is coffee good or bad? I mean, when is enhancement positive? And you know, that answer comes down to people arguing what they feel about, but we need some more of an objective base for that, I think, to really address these things. And you know, I think there's some ideas we have about that, but you know, we need an objective base. It can't just be whether you think it's good or bad. And that's where some of the ethics and the frameworks are going to be important. It's not the individual, society, it's the universe. I mean, we have to sort of look at good and bad on what scale as well. So,
but how you suggest you do this? I mean, at the moment, we live at the time of this horrible pandemic, and you see, yeah, we can't agree about is the vaccine, getting a vaccine good or not? You know, so, and this is a relatively simple question compared to the questions that we are posing here. What are your suggestions?
Yeah, but that question was made very complex because it was confounded with another simple thing, that I should have individual freedom. Right? I mean, so we can look at different views, and of course you should get the vaccine, you're gonna be healthier. But when it's forced on you, people say, well, you can't force me to do it. And it's total BS argument in some ways, in my view, but not that I should say that out loud. But what's the objective way of getting at that? And so I don't have an answer for you. We're starting to play with ideas that, you know, Carl Friston has published a lot about the idea of free energy and pieces like that. These are objective measures about how entropy and energy go forward, and they're can something like this be applied to what we're doing? And I think there's an interesting idea of the entropy of society. What is that? How do you measure it? What is the energy society takes to get there? These kind of questions can give us objective measures. They're going to be grand scale. They're going to be things you look at over time. But, you know, I think we can get away from what this sect believes, this religion, this government, you know, but actually look at what is an objective way of measuring it. If we can ever figure that out, it's going to be a game changer. Otherwise, we're going to continue to argue by whoever's got the strongest voice.
Oh, I'm actually quite a pessimist there, I must admit, Dustin. I think we will continue arguing for a very long time, unfortunately, as we have been arguing a long time about the use of embryonic stem cells and now iPSCs. So, Jack, that is your field. What is your view on that? You remember that there was still this huge opposition about the use of embryonic stem cells, and we can all understand this. But then, you know, iPSCs came about, and we all thought, okay, this is going to be ameliorate the issue of embryonic stem cells quite a lot. But it seems that we were not completely right about it. So what is your view on the ethics there and the current problems that we're experiencing?
Well, there's some interesting issues there. If I might just very quickly refer back to what Dustin's been discussing, the issues around stem cells are so much easier than the issues that Dustin's just raised, because I think the issues we've got to deal with, we have machinery to deal with them. The thing that really worries me about what Dustin's been alerting us to is we quite clearly don't have the mechanisms to deal with it. He's made a couple of cryptic comments about Facebook, which I totally share, let me say. And it's quite clear, as he implied, that you cannot trust these commercial organizations to deal with this stuff in the way we would like. But the problem is, at the present moment, these things are almost totally in their hands. Now, in stem cells, that's also true to some extent, and perhaps we can come back to that if we're interested in it. To deal with the specific issue that you raised, I think it's an interesting one, because it's never totally been settled. So the current situation, just to fill everybody in, is that in some countries, particularly those with a stronger religious background, the use of ES cells is still somewhat prohibited to different degrees. Some countries have no legislation, interestingly. I mean, Ireland, I think I'm correct in saying, more or less has no restrictions, which you might imagine slightly surprising, given France, Italy, Germany, countries with a Catholic tradition do. In the UK, we have a very regulatory system, which is, I know the regulators themselves don't like it being called permissive, because they don't see it that way. But in fact, there isn't much that it inhibits in terms of progress towards biomedical uses of embryonic stem cells. And of course, we have the UK Stem Cell Bank, which is really unique and showed a great foresight. Charlie's online. I mean, the MRC and the BBRC had incredible foresight, I think, in setting that up back whenever it was, 1980s, I believe. At some level, it's okay for different countries to have different regulations, but there are problems that are going to arise, and the simplest one to see on the horizon is when the medicines finally come through. I mean, Viviane gave this brilliant talk, didn't she, yesterday, about the stuff that's going on in Parkinson's disease and the stuff that Blue Rock are doing in the US and people are doing here in Europe and also in Japan. Really exciting stuff. I mean, I hesitate to say this because I've been wrong so often, but I strongly suspect this is going to work. I mean, the science now is so solid, and they've worked through so carefully the implications of it and the nuts and bolts of how to go about it, that I strongly suspect this is going to work, and that's going to be a great breakthrough. But it's going to immediately focus attention on the ethical issue that you've just raised. So let me pose a problem that the European Medicines Association, the EMA, the European Medicines Regulatory Agency, are going to face very, very soon, and that's going to be this. Data is going to be presented to them, let's say in Europe, let's say in the UK or Scandinavia, saying we have this medicine, it works, we want regulatory approval. And the EMA are going to have to give it, okay? And then all these countries that have forbidden ES research are going to have to look at that and decide, do they make those therapies available in their countries. And I mean, Mikhail will have a view on this. My strong impression that countries like Germany that have backed off allowing research, I mean, you can do ES research, let me say, in Germany, but with more severe restrictions than in the UK. I think patients are going to be clamoring for those medicines, and very, very quickly regulators in Berlin, in France, in Italy are going to have to change tax pretty damn quickly. But that's going to be an interesting issue.
So
I would say, in terms of regulations in the UK, I think one encouraging sign from our side is the Regulatory Horizons Council, which is a non-governmental council that looks at emerging technology areas, are currently doing a deep dive on neurotechnology, where they're looking at regulations, but also ethics. And so I think there is an awareness across government and elsewhere that this is an area that needs to be looked at now. It's not something that in the future we should be thinking about. Across government, there's a human augmentation working group that again is trying to set out some ethical principles in collaboration with the Royal Society. So I think it's encouraging that there is work going on now to be thinking about some of these principles.
Yeah.
Absolutely wonderful. Thank you for joining, Charlie, on this. I was just I just wanted to ask you, where do we stand and where do we see the highest interest regarding neurotechnologies in the UK? But it's so encouraging to see that the government actually already is sitting down and thinking about it a little bit. It's not really ahead of a time. It's already kind of lagging, but not lagging maybe too much with all of these regulations. I actually, there was a question, something when pain popped up. Maybe this is not really correct, but Dustin, when we talk about sensory experiences, I mean, it is true that losing sensory experience because of spinal cord injury or loss of a limb it's terrifying. It's life-changing, for many people actually devastating. And but what people actually, at least what I have seen, people who lost their limbs or been spinal cord injured, what they report is even worse is of either phantom pain or allodynia, things like that. So can your work, for example, also help along those lines? Can we actually dampen the pain, like we can increase or restore the sensory perception, but can we actually kill the pain in a way?
So I'm going to answer this with more of a observation than a detailed study, right? We haven't powered the study for this yet, but I will say that in our instance, so others have seen this as well. A few other studies have been going of showing that sensory will restore. It depends on the mechanism of pain, right? So there's a couple different ways in limb loss, for example, that you get pain. One is true neuroma. So nerve grows out and then you touch it, the pain fibers fire. It's actual signals coming back that are pain signals. There are techniques that are being developed. There's a couple companies that haven't yet, are very close to getting FDA approval, that can damp that input, right? So in the periphery, you can put input, you can actually quench the activity being transferred to the brain, and that cures the phantom. But there's also this cortical misorganization, right? When the sensory input stops coming, the brain fills it in. It's often following traumatic limb loss. So a lot of times the phantom pain is highly related to whatever they were doing when they had the limb loss. So one of our subjects, for example, was holding tongs in a punch press that came down and took off his hand. His pain, as he would describe it, is he could never open his hand. So the image of his hand, his phantom, was always clenched. And what would happen with the pain, it would be like a vice and just pushing his fingers through his palm is the way he would describe it. And he couldn't do anything about it. As soon as we started getting stim, one of the first comments he made was, "My hand just opened up." So because you start to give this sensory feedback back, there's something for the brain to now connect to and more of the hand being there. And so he has not had phantom pain since. The second of our subject lost his hand in an industrial shredder, right? So took his hand off and his sensation was like a nail being driven through his thumb, he would say. It's gone. So absolutely it can make an impact because this reorganization, this cortical misorganization, the memory of what you've had, all these type of things. So there's lots of different ways. People have even shown with VR virtualization, viewing or mirror therapy. It doesn't work for everybody, but if you're missing your hand, but you see a hand and you kind of move it, it's the brain reconnecting with there being a hand and getting rid of some of those misorganizations that relates to a lot of the phantom sensations that people have. So most definitely. So I hesitate because we haven't powered it, but yes, we see absolute results from that perspective. And us and others. So yeah, 100%. And the relationship with the hand, we haven't seen that the phantom goes away. So it's very interesting that our subjects will talk about, you know, I have my phantom, but when you turn your system on, I feel my hand is different. I recognize a difference between the two. So we're still looking at, will the hand disappear if they're using it every day at home, 100% with the phantom, where they see a touch and feel a touch, will the phantom go away? The last thing I'll say about that is the other close part to that is if somebody is not wearing a prosthesis and not having sensation, their phantom is often at the end of the residual. So their fingers will be on the end of whatever the arm they have. As soon as they put on, a lot of times just putting the hand on from a visual will allow it to extend out. But we see this even better when we add sensation. It'll actually go out and align with the prosthesis. So there's a lot of changes to the phantom as the visual tactile synchrony comes together. Again, remember, our view of reality is an integration of all the senses. And so as they come together, as our agency is starting to see it work on a representation, what becomes a more interesting question about that is, could you embody, or would your phantom, would you begin to embody something that's not anthropomorphic? So if it's not a 5-fingered anthropomorphic hand, but you move and it moves exactly as you intend it to, it touches something and you feel that touch, you begin to embody this non-anthropomorphic sense of self as well, which is really fascinating research that is opening up from this, but we don't have a lot to add to that. So, you know, you're plastic, we know that. And now we can play with that by having an input. And so you can kind of change perceptions and get back and forth to where it is. So absolutely, lots of interesting things there.
Wow, it's, you know, I think I could just ask questions on my own without even looking the audience's responses. Let me ask you something that I have seen previously in the question feed here. When we're talking, and I think we already touched a little bit upon this, when we talk about all of these technologies, of course, when there is source of funding, which many people we know immediately will flinch when they hear about it. I don't want to name the agency per se, but it has something to do with military use. What is your view on that? I know that, you know, sometimes when I hear it from my colleagues, oh, funded by them. No, it must be something, you know, it's not very good. So what is your view? Because there are lots of war vets, for example, loss of a limb, brain damage, spinal cord injury. So
that's why it was funded. Yeah, so it was funded because of people coming back with missing limbs, right? And if you look at the history of prosthetics specifically, it actually has only progressed during war, right? So cause and effect is the question here, but you know, lower limb prosthetics actually made huge advances during World War II with landmines, people losing a lot of limbs from the landmines. And the same thing now, upper extremity, because a lot of IED and the things, people would now survive because you have good core protection, but you come back missing 3-4 limbs. I mean, it's horrible. We think about somebody losing their arm. What's coming back from the military is people with 2 legs and an arm missing. And so, you know, it's one thing to lose a hand, it's totally another to lose both of your hands, right? I mean, so sort of think about that. So there was a real need from the returning vet. So that was the initiation for the work. The stuff I talk about that goes beyond that gets back to the original statement of technology itself is neither good nor bad. So you can certainly imagine things, for example, explosive ordnance disposal. So people that go into the field to try and disarm IEDs, right? We actually visited a group here, and I asked them, I said, you know, how many times did you go out in a week? I was thinking 4 or 5 times a week. These guys are going 12 times a week into a day into the field, where they have people watching how they're doing it, shooting at them while they're going to try and dispose this bomb, seeing what they do, and then resetting it to try and get around whatever they just did. And so you can imagine, and I kind of asked, what's your injury rate? Half of them die. I mean, it is the most dangerous job in the military. It's not the frontline infantry at this point or something like that. It's somebody that's going out for IEDs. If I could remove them from that environment, changes the game. So, you know, but now sending drone warfare, that's already an argument. You know, there's psychological elements for drone warfare where you're shooting from a distance. These people have psychological elements to it, right? And it's weird to think about the humanity of war, right? But if you actually are there looking in their eyes when you're doing something, or imagine just for the hell of it, right? This is a total thought experiment, but if you felt what they feel, so thinking about a shared experience, then what would you do? I mean, and even go beyond, go again, not to wane too far, but go before the war, right? What if you experience their life? Most of us have these differences because we don't know what other people experience, right? Think about the most powerful movies you see of other cultures. You feel like you're there and you start to understand their environment. Imagine if you could feel their environment, you start to get a reality. I mean, think of the value of not so much movies, but now 3D representations where you can actually understand what it's like to live like somebody else. Maybe empathy changes and we can actually avoid that war part. So yes, there is always a nefarious need for these things, and I agree with the concerns. And hell, when I took the money in the first place, it's like, where are we using it? So you have to balance. It's the same thing as always. It's not inherently good or bad, and there are a lot of places where I think it brings value on the plus side, but we have to protect against the negative. And that's the ethics thing. We have to build it in. And I'm going to say one other thing to your earlier comment, Marija, that you talked about, like, people were going to continue to argue, I'm a little too Pollyannish, I'm too optimistic. I actually don't believe we'll ever stop arguments, right? I think this goes back to the original notions of the philosopher king or something like that, right? It's not so much that everybody has to understand the arguments. It's more, I think, about the fact that we have to know that somebody does. So if you're on a ship, you trust the captain's navigating. I may not know it, and it may look funny, and I may argue about how he steered the ship, but at the end of the day, I trust he has the maps in the background, whether I can read them or not. I think what scares me is if there's nobody in the helm, right? So the trick is not so much because it's complicated and people are going to stop arguing. I don't think that's ever going to happen. The question is, are we making sure that we actually have something to guide by? And if we do, then you keep people on the rails, right? So the society can, I mean, I don't mind the churn, right? It helps. But you just have to make sure somebody's actually steering the boat in some way. So I don't ever expect we're going to stop arguing. I'm not that naive. But I do think it's important that there is at least a group that is thinking more broad scale. And, Asimov's foundation, right? That if you've got Apple TV now, again, literature leading the way, the foundation is essentially, the basis of that book series is on this idea of an underlying equation nobody understands. So, I mean, it's kind of these key ideas. I don't want to give up because people won't get it, but we have to keep pushing on that and then figure out how to manage the arguments.
And exactly. And, sometimes arguing is still fine. Mm-hmm. Bigger problem may actually occur when we stop arguing. Yeah. I'm actually worried more about that because then nobody's really in charge of anything. But it's really interesting how you started working on all of this and saying, war vets and we need to help. And on the other hand, we have mental health disorders and various conditions from depression, PTSD, and we have heard yesterday that, for example, we can use virtual reality, probably even DBS, to help to erase some bad memories. So maybe, Michal, you could comment on this. How is it possible to use such technologies like TMS or DBS to help there?
To help to make to re-experience traumatic experiences, or what do you mean?
No. So, for example, Lucia Velmaja yesterday was mentioning, but it was more about virtual reality. That these technologies can be used to help in, for example, in psychotherapy, to help people experience severe PTSD, for example, to in a way ameliorate or even erase bad memories. In your experience, how would that work? Where are we standing with that?
Yeah. So it's still at the beginning, but it depends on the field. So it's used more and more, still not very much in clinical routine, but in let's say studies in psychotherapy, for example, treatment of phobic diseases, mm-hmm. where it's relatively easy with a spider. I mean, they don't need much. But if you think about high places or something like this, or if you think about flying, I was frightened about flying for various reasons many years, and then my solution was at the end just fly, and I was in bad mood and thought, okay, if it comes down, doesn't matter so much at this time. Had some benzodiazepines, did a long flight, everything was fine. But and they have also these flights, at least in Germany, some psychotherapists are offering this, and they have a plane full of people being afraid of flying. So what it makes it easier in situations where people are afraid of specific things to experience this and to have this kind of exposition therapy, which is at least as efficient as when you have real exposition, if the virtual reality environment is okay, because people really feel, I think the crucial thing is that you feel that you are in it. As long as you have your distance and it's all not real and whatever, then it's not very useful, because you need to develop, you need to experience the anxiety and these things. But as long as when it's realistic, then I think it will be an advance, because it makes things more economic, and currently I think that's a worldwide problem. You don't have enough psychotherapists, so people are waiting for 1 or 2 years and whatever, and of course this is has a relevant impact on life quality. So there I'm relatively optimistic, and things are going on and things are under development. Will still be technically optimized, but that's relatively advanced. And of course for PTSD this will be even a greater thing, because you still, so if you need exposition, you still can get to a plane, but you won't re-experience your trauma in reality. If their exposition is also a thing to work this through and to get over the peak of anxiety and so on. And so this might be something for the future, but I'm not a specialist in this. I'm not so sure how far they are in it now. Yeah. And you can combine this with other interventions. So DBS might be one thing. We're having a large consortium projects about extinction learning and phobias, and then of course we combine this extinction as a model for exposition. We combine this with brain stimulation, try to boost the efficacy of the therapy and to stabilize the effects. I mean, that's another thing, that whatever you do currently, it's often so in many patients it works well, but there is a relevant group of patients who have effects which vanish after a while. This exposition is still great, but it doesn't work in everybody. And we know, at least physiologically, you need specific protocols to stabilize things. So you need to reapply this, and when we induce plasticity or modulate plasticity, so we form a new memory for extinction then with if it's with DBS, if it's with non-invasive brain stimulation protocols or with other things, this would be, of course, great. That you don't have to do this again and again over the years, but you do it once, you have a stable memory, and then that's it. So working on it too, but that's also still science and not routine clinical application.
Yeah. Thank you for this. You again reiterated the fact that kind of something that we discussed yesterday, that it can really, first, we don't have enough psychotherapists and everything, but it can also speed up the process, which is incredibly important for the individual to kind of get rid, if possible, those bad memories and bad experiences as soon as possible. And Mitul, just in case, I mean, if you really successfully develop this kind of extinction of bad memories, like spiderophobia, how I like to call it, just please let me know. I would love to get rid of that. I know it's trivial compared to everything else, but you know, I see a spider and I'm out of the building in a way. It's really bad, you know.
If it doesn't work, I send you a spider, okay?
Charlie, when we're talking about all of these things, I think you have probably the best overview of UK environment. So where we are standing, you know, I don't want to really talk competition here, Europe versus US, or even worse, UK versus EU. But Michael is laughing there. But you know, this is a big topic these days, or the last few years. What seems to be most interesting when we look into new neurotech companies, what seems to be kind of up and coming, not just well established, but really, I don't want to really say kind of financially most lucrative, but what seems to be the most interesting from the funder's perspective?
Yeah, well, I think perhaps if we start in the US, the companies that are getting a huge amount of funding now are wearables, so they're probably sleep trackers or those kinds of companies. So they've had, you know, millions of pounds, millions of dollars investment. In the UK, I think we've got a wide range of technology areas that we're doing. I think one of the areas that was just mentioned then was TMS. So Maxim, of course, really the pioneers of TMS. It's getting rolled out in the NHS at the moment, so that's definitely a key strength of ours. But I think perhaps one strength that we can look to is also other industries around us. So the creative industry, for example, in the UK is a real strength of ours. And I think collaborating between people developing brain-computer interfaces may be there for stroke rehabilitation or other things like that, but also collaborating with the creative industries could be a real bonus for the UK. And of course, we've got a fantastic pharmaceutical industry as well. GSK obviously are getting very interested in neurotechnology with their spin out Galvani. So I think there are lots of strengths of the UK. We've touched on regulation. I think that there's perhaps with Brexit, there might be an opportunity for us to have a slightly different regulatory approach. Who knows? But we'll have to see how that turns out.
Actually, I think that all of us from the EU side, kind of the channel, we are all very, very curious to see how that will develop, you know, and what will happen. Really.
If regulations become more reasonable, then it's in the EU, just let me know. In the EU, it's of course it was always bureaucratic, but we had always this problem. But I think this was also a problem in UK, and it's also a problem in US how they rate studies now. So I think in the US now whatever you're doing with brain stimulation, it's in itself a clinical trial, which by law, and so all the non-invasive brain stimulation guys at least have lots of problems with this, because these are quite other kind of studies you have. So it's over-regulated at the end for this kind of preclinical stuff we are doing and exploratory studies. And now they start to fiddle around with CE certificates and all these things. And it's not only the laws, it's also the people interpreting this. So there was a huge discussion about what's the medical products law, and of course there's comments to the law that if your scientific interest rules you're not a company, then that's not medical products law studies which you are doing, because they have another reason, and this law is another basics. But sometimes regional authorities don't know this, don't understand this, ethics commissions don't understand this, lots of fuss. But I think that's a common problem in the field currently, and if this gets more reasonable, this will be a big benefit. Short additional commentary, what I think not only in the UK but in general in the field, and this not only applies to non-invasive stimulation but to many interventions. I think the future is multimodal therapies, and so we were working. We've done a lot of pharmacological studies combining with brain stimulation, and if you do this in a reasonable way, if mechanisms are shared. So, for example, if we combine TDCS with serotonin uptake inhibitors, you get a huge boosting effect on plasticity, and there are pilot studies which show that combining these approaches is significantly more efficient than each approach alone. And so I think the thought is there for many years. It somehow never worked in larger style because of don't know which problems. But pharmaceutical companies are getting more interested and to tackle problems from different angles. I think this could be really could be something for the future, which could be very relevant.
Could I make a comment on that, Marija?
Sure, absolutely.
So just two comments on the regulatory thing. One is obviously following Brexit, we can diverge in regulatory terms. I suspect that will be less prominent than one might imagine for two reasons. One is the UK is a relatively small market compared to Europe, so pharmaceutical companies are not going to want to work to two different standards. And the thing that exacerbates that, and obviously I realise the politics are difficult here, I don't want to get into it, but given the Northern Ireland Protocol, medicines in Northern Ireland actually come under the EMA now rather than the MHRA in the UK. It's a mess, but that means divergence, I think, is going to be less easy than might otherwise have been the case. just to follow up on Michel's comment, with which I totally agree. I mean, regulations are desperately trying to catch up because medicine's changing so fast. It's the usual thing. Technology moves and the regulators have to try and catch up. I mean, there's a couple of drivers there. One is simply personalized medicine. You know, if you're producing a medicine for a single person, as some of the advanced therapies really are now, it makes no sense. I mean, how do you do a clinical trial when every patient's getting something different? So that's a real challenge. And the one, again, Michel's just referred to is the regulations around devices really need to be revised. Combination devices, they quite often have a device component and a biological component, be it cells or whatever, are going to become more and more prominent, aren't they? And the regulations around those are really not up to scratch. Currently, the two components, the cells and the device, have to be regulated independently. And obviously that doesn't work. So there's lots of challenges to regulation, and it's going to be interesting to see if regulators can catch up, particularly as, if I might, my final point, particularly as there's so many companies out there, particularly, Dustin no doubt be aware of this, particularly in the US. I mean, in Europe, regulators work pretty tightly, and these sort of private enterprises that come up with, you know, fake therapies get closed down pretty damn quick down here. In the US, it's an enormous problem. You know, direct-to-consumer medicine is a way of life there, and I'm very aware of it around stem cell companies, which the FDA have struggled for years to try and close down, and really, well, I hate to say failed because that's not fair. They've succeeded in many regards, but there's so many rogue companies out there selling untested and invalid medicines over the counter to vulnerable individuals, and I'm desperately sad to see some medical schools, some, you know, high-profile medical schools in the United States joining in. It's really distressing from my perspective. So the regulators have definitely got to catch up.
Well, and
Just
to add to this from the U.S. side at least, and I think it's global actually, that the one thing that, and I won't mention any large companies obviously again, but you know, the requirement of data to the public is zero. Right? Anybody, I mean, it's horrifying to me to read that people read and listen to. And so you can buy just about anything, I mean, the idea that people have forced in the US doctors to give them ivermectin because they don't want to take a vaccine is mind-boggling to me. But that just shows you the problem of the lack of information are out of control from that. So it's not just regulatory from that perspective, right? And somebody will try to make money off of anything from somebody, right? So the regulation schema that we have is probably broke relative to the social environment we have now. That's the bigger problem, in my view, in many of these things. And I don't know if we have time to talk. I'd be curious. You know, I was going to ask a question. It looks like maybe we're out of time, but you know, we've talked a lot of stuff here. But what I'd be thrilled to get your input on, and I've been struggling with, is all these questions need the ethics in front, right? We are scientists that do this stuff, and we say this isn't ethical. But I've been finding some traction, but difficult to have the dialogue with the ethicists. Right from the beginning, right? I mean, how do you have an ethicist sitting in your lab? No. Like, when do these discussions happen? And are they real, or are they just tangential when we're interested at a conference? So I'm curious if any of you have really engaged those discussions outside of your own machinations, or are we really engaging ethicists and people to think about these things at this point? Or, you know, how does it work in your groups or across the UK, EU? How does this stuff work for you?
I could perhaps address that, at least from my own experience. So the answer to your question simply is no, we don't engage ethicists right up front. What we do have, and it sort of bridges the gap, but only sort of, is we have organisations, certainly in the UK and more internationally, that try and address these questions sort of independently. So one is things like the Nuffield Foundation. You'll be aware, in fact, I was on a team that reported on novel neurotechnologies a few years ago. We tried to raise the issues. We have an input into government and through, you know, the Medical Research Council, etc. That's another important avenue. The other avenue that I think works to some extent is some of the sort of learned societies who try and get involved in this. So probably the best, for example, in my own field in stem cells, probably the best source of guidance on ethical issues is the International Society for Stem Cell Research. In fact, they've updated their ethical guidance this year on some of the issues that we've been talking about around ES cells, about use of, I mean, you'll be aware of this issue around disease modeling using brain organoids and whether they're conscious or not, and so on and so on, all these issues, which actually points out one of the slight downsides, Dustin, of what you were suggesting, getting ethicists involved too early. I might hesitate to suggest they sometimes go over the top.
Yeah, this is part of the challenge.
They can sometimes get a bit out of hand quite quickly.
Yeah, we end up contemplating your navel, which also isn't useful.
Yeah. So I think there are sources, but your point about getting involved more at the front end, I think, is well taken.
Yeah.
I'd also say that the Royal Society have started looking into this area a lot. They've run public consultations to really understand the public view of this area, as well as just which will hopefully inform policymakers. And I think, and also something useful that happened recently, Defence and Security Services approached us to run a security and ethics workshop to actually try to bring together scientists and ethicists. So there is work through government that's trying to make this happen to some extent as well.
Yeah.
Cool.
Really excellent. And I think that we will probably stop here and kind of having this freedom of science versus ethics, or kind of combining them, and when to start this dialogue as a food for thought for all of our audiences. And we actually, very quickly, for me, it all went very quickly with such excellent topics and speakers. We are now at the end of our event, and I would like to ask Mitul to join me in with closing remarks. Before Mitul takes over, I would really like to thank all of our speakers. It was really wonderful of you that you joined us and accepted these invites and gave these wonderful talks and told us about your research. Thank you to also big thank you to all our wonderful audience for keeping up with us for 2 days. And Mitul, stage is yours.
Well, thank you very much, Marija. Thank you very much to the panel and all the speakers. It's been a totally fascinating whirlwind ride through various neurotechnologies. We started yesterday really with the human, whole human, let's say, and we heard from clinicians, we heard from scientists about the technologies they're developing, and then we moved into the cellular space and we could learn about the technologies developed there and how they're moving so rapidly, and they're becoming much more advanced, and possibly, as we heard from Viviane Tabar, living up to the promises that we've had for so long, which is fantastic to see. And then today on day 2, it's just been even better, if not equally good. And you know, really seeing not only the panel speakers here, well, Jack, of course, and Michelle spoke yesterday, but hearing the panel speakers and the speakers from today, from Dustin, you know, from Ed Boyden as well, really working at the cutting edge, doing something which I think I'd say we couldn't imagine recently, but Dustin already corrected me before I even thought of saying it earlier. But many scientists couldn't imagine would be a reality now, and seeing it happening is just marvelous. So we've you know, we've also learned a lot on the ethics. I was involved very much in the smart drugs debate, working in psychopharmacology, and a lot of the discussion on the ethics really echoed the discussions we were having maybe a decade ago around drugs to enhance cognition, and the ethics and the morality even of having these pills available to people. And I mean, we did have some concrete examples there, like stimulant drugs being taken by students to hope improve their exam performance. But I would argue, as Jack was saying there, that those debates were probably too early, because those examples were there, but they really didn't grasp really where the ethicists thought we were going. And in a way, the debate was going too far, too fast. And myself as a neuroscientist felt a little bit out of sea. And my contribution felt like I was pulling people back into reality. I think one of the differences now is that we can have these debates with a very clear sense of what our reality is and what our proximal reality might be. And if we were to maintain the debates around these areas and then separate out the extension around where we were going, they could be much more successful, much more useful, and not feel like free thoughts happening over and over. So I feel like I do feel like I could really benefit from some of those drugs which were hypothetical in order to keep up with where the field is going. I work in brain imaging, and I've always been impressed by Alessandro Gozzi's work and seeing how he is combining different methodologies to try and give us different, I suppose, biotypes of autism from the animal perspective, and then moving into the human to validate. It's the opposite of what the rest of the field is doing. It's just amazing. It's stunning. So I'll probably need a lifetime to catch up and learn all of these techniques so that I can make a difference. So I'm going to leave this meeting feeling very humbled, very small, very impressed, and delighted that surrounded by a world of expertise, and just have to really pull it all together to see how we can work together to benefit the whole field. And I think the potential beneficiaries of these techniques and technologies would be delighted to hear what's being said in these meetings. And of course, their involvement, as we all know, is vital in shaping the discussions and shaping the technologies and how they're implemented. And so I'll leave on that note, really. I'll leave us thinking about the potential beneficiaries of these technologies beyond the knowledge that neuroscience will gain towards the benefits that individual people might experience. And thank everyone once again. And finally, thank Marija for your expert sharing. It was almost as enjoyable as the talks and deserves a round of applause. Yeah.
Thank you, Mitul. It was a pleasure. But it's easy to be a chair with such wonderful speakers and wonderful topics and questions from the audience. Thank you all again.
Yes, thank you. Bye, everyone.
Thank you.
Bye, everyone. Until next time.